So once we get moving, we are going to be traveling upslope. We're at the northern end of Lang Bank, the northern side of Lang Bank. So we'll be traveling to the south. And Lang Bank is really big. It's 15 kilometers wide, basically, and it's very, very shallow at the top. It's about 30 meters.
Yeah.
So we're going to be kind of slowly moving up the edge of the bank to about 300 or maybe even 250 meters, which is about the top of the operating depth for our ROV. Oh, look at the fish.
We're starting to see here now some fawn already. This couldn't quite see exactly what it is. Possibly one allosauride.
And what we're looking at on the seafloor right now is probably mostly carbonate sediment, perhaps with some hard bottom areas. Obviously has some bioturbation in it.
Yeah, and it seems to have some biogenic material as well, the darker portions that we're seeing now. We'll just wait to have zoom in to be able to say a bit more about it.
Thank you very much.
So we will now start on our transect.
As a reminder, we're not going to be collecting samples today except water samples.
Exactly.
Because we're within 12 nautical mile boundary. Did you know that St. Croix was the largest of the U.S. Virgin Islands?
I did not.
Yeah. So we're not really going to encounter any very steep slopes, I think. I think this is going to be, we're going to move up slope slowly and surely.
Still hopefully seeing some rocky outcrops or areas of harder substrate, which typically then host dense communities, especially with corals and sponges.
And right now we're a little bit below the optimal depth to find some of these commercially important fishery species, right?
Yeah, that is true. So typically they will occur between 50, 100 and 500 or 600 meters depth. That's typically where most of the local fishing communities will be operating on. This is probably an allosauride fish that we're seeing here that has this very long, wavy fin. But of course, surveying these deeper areas is very important because sometimes it's during these exploration dives that we actually see range extensions in terms of bathymetry for some of the species. So it's not uncommon that some fish species look for deeper areas, for instance, to spawn. These areas can serve as nursery areas, with the adults then returning to relatively shallower waters. So it's always good. Oh, and we have a first brittle star of the dive. A couple of them, actually. It's actually quite large as well. Mm-hmm. Very long, relatively long arms.
It's also fully splayed out. A lot of times we see them. Mm. This is good. This is really big. That's like 25 centimeters across from arm tip to arm tip.
Oh,
did you see that?
Yeah.
That fish just dive bombed the bottom of the sediment.
Perhaps because of the lights of the ROV, maybe. We don't know. Oh.
What kind of fish is that?
I really don't know.
Other than very shiny. Very, very shiny.
It's like 10 centimeters long.
Oh.
Whoop, there he goes.
Well, he caught it for a moment.
And I think we have here also black coral, possibly in the Stichopathus genus. They often have this coiled appearance. I think we've seen actually these on our previous dive as well, these orange Stichopathus. Yeah, can we take a look at this? If we can have a snap zoom, that would be great. Yeah, and we can see here the central axis, this very dark orange color, and then with the polyps on both sides.
Very cool.
Very long. It's very long tentacles there. Oh, beautiful. Yeah, it looks very similar to some of the individuals we've seen in our previous dive, actually, on the monoblock.
Oh, nice view.
Great view. Oh, we have there another fish to the left.
Another coral. We're all kind of wandering off the left edge of our screen right now.
Oh, and now a shrimp as well. It's actually a large shrimp. Let's have a look at that. Interesting. It seems to be.
Seems like he's working very hard,
Yeah,
not
to
going
get
anywhere.
exactly to get.
It's almost cartoonish running in place.
Yeah. I wonder what that behavior is for.
Yeah, that's a good question. I'm not sure. I don't think we have a lot of current here in the right now. Yeah.
So if it's a light current, and so this shrimp is not necessarily fighting to stay in place,
No, indeed.
it must be doing something else that has a behavioral component.
Yeah. Yeah, and we can see that this is it's often these type of behaviors that cause these trace marks that we often see on the seafloor. So right now we might not be knowing exactly what it's doing, but
Not a clue.
and it seemed to have just settled down now, and that's very interesting.
Looks a lot like my dog when it was trying to find a place to, lay down. They like
And
to
just
circle,
chill.
and then they sort of like, mess up their cushions, and then they just sit down and chill out.
Look at that.
Yeah, when it started, it was above this little lump of sediment, and now it's kind of slightly behind it. I wonder if the motion was
To create that lump?
like you described, Deb, like organizing a spot to be in.
But just as a reminder, I'm a geologist, so I'm just speculating.
Sure, yeah.
It is very interesting. Oh, didn't like that area for very long.
No.
What do shrimps eat?
So they eat a lot of small particles that are either on the seafloor or on the water column. So they can eat other small crustaceans, small particulate matter that's also can be on the sediment or on the water or in the water. Is there a rock there to the left?
That's what I was wondering.
Ooh, and that was a very fast fish. Yeah.
Sure. We just do a quick snap zoom.
Yeah.
Oh, yeah.
It actually looks like, I don't know, broken.
It looks like a piece of coral reef.
Yeah, exactly. It looks like a broken piece of coral, or it looks very biogenic.
You think that's a hermit crab on top?
Yeah, there is a small hermit crab in its shell.
That looks less like a rock now that we're close to it.
Yeah. I'm starting to think it could actually be a sponge skeleton.
I was waiting for you to say that, because that's what I think it is too. But you're the sponge expert.
Yeah, it actually looks more like a sponge skeleton now.
Yeah, it does.
And a rather fibrous one as well.
Cool.
So it probably rolled down. So it wouldn't typically occur here over the soft sediment, but it probably just rolled down from the shallower areas.
That would have been quite a wild ride if the hermit crab was on it the whole time.
And another one of those, I don't know, probably again a sponge, and we have a crinod over it.
Mm-hmm. And then another black coral to the left?
Yeah, to the left. Possibly again another one of those Stichopathas we've just seen.
Is there something on it, though?
Yeah, it looks like something is growing on it. Yeah, sure. Thank you. And actually another one there to the left. So there's a few of these Stichopathas black corals here in the area.
Oh, it's a brittle star.
Oh, it's a
Holding on
brittle
for dear life.
star. Yeah. But it's rather curious because we don't often see these brittle stars growing on these black corals, at least on the Stichopathas. So that's very interesting.
But they do get higher up in the water column.
Yes, that's true. So we've seen some of these usually associated to Metallogorgia, another type of coral, the golden coral. But in our previous dives, we haven't seen them associated to Stichopathas. Mm-hmm. So that's a new observation. right there.
Good morning. It sounds like we have somebody joining us on the phone line. Maybe not.
We just heard a beep here on our
like it.
Yeah, it certainly did. Yes.
Oh, there's another fish to the left. Maybe it's a fish, maybe it's not a fish. Oh, maybe it's nothing.
There are a few very small ones also on the water column, actually.
There we go.
Oh, there we go. Oh, they are very fast.
Okay, we're going to take a moment here and do some introductions.
Hi, everyone. This is the pilot currently, piloting D2. I'm from the Global Foundation for Ocean Exploration, or we're known as GFOE. We're made up of engineers and videographers. We're responsible for the vehicles, not just for building and maintaining them, but also operating them, and then also all the amazing video products you see today. I'm a mechanical engineer. I'm part of the team, and we'll just go through the rows real quick. Sitting to my left.
And I am to pilot's right. I'm Andrew O'Brien, currently sitting co-pilot. I'm a software and data engineer, and I'm also with the Global Foundation for Ocean Exploration.
Good morning. I'm Brian Doros, broadcast systems engineer with the Global Foundation for Ocean Exploration, sitting in at video and broadcast control.
And my name is Anna Sagatov. I'm sitting in the clipping station right now, and I'm with Global Foundation for Ocean Exploration.
Hi, good morning and buenos dias. I'm Joana Xavier, and I'm a researcher at CIMAR, the Interdisciplinary Center for Marine Environmental Research at the University of Porto in Portugal. And I am here with the NOAA Ocean Exploration as a science lead for biology on this EX2206.
I'm Deb Glickson. I'm with the National Academies of Sciences, Engineering, and Medicine, and I'm the geology lead for this expedition.
And last on the back row, I'm Thomas Morrow, Expedition Coordinator for NOAA Ocean Exploration. Thanks, everyone, for introducing yourselves. If you're just joining us, we are just a few miles off the east coast of St. Croix, diving on a feature known as Lang Bank. It's of particular interest for fishing communities in and around Puerto Rico. Joanna, if you would care to introduce our dive to the Spanish-speaking community?
Sure. So I think I will make a small, just a brief summary of where we are in Spanish. Hola, buenos días para quien nos sigue. Pues estamos hoy haciendo el séptima exploración de esta campaña EX2206 de la Voyage to the Reach con NOAA Ocean Exploration. Y hoy estamos aquí a como unas 9 millas náuticas de St. Croix, que es conocido como Lang Bank, y esta es una zona muy importante en términos de soportar comunidades de peces, muchos de los cuales de importancia comercial y por tanto soportar también una economía local aquí en St. Croix y Puerto Rico. Hoy en nuestra exploración seguirá el slope del Lang Bank y vamos a hacer un transecto entre los 700 y los 300 metros de profundidad, donde esperamos poder observar con algún detalle y caracterizar las comunidades de peces, pero también las comunidades bentónicas y el hábitat que soporta estas comunidades. Y muchas gracias por seguirnos y seguiremos dando pequeños sumarios a medida que vamos avanzando en este mergullo de exploración.
Thanks, Joanna. So we're coming up on what I would characterize as our first change in the slope. A little more variety in the features here.
Whoa, what's that thing?
It's a very large. Is that a crinoid in the distance?
Yeah, and we have actually also here some cans, and we've seen also a glass bottle earlier on. And again, I wonder if that's actually a sponge and not a rock underneath that.
I think it's a sponge, but maybe I'm wrong.
Yeah, that looks like a sponge, actually.
I mean, last time I was wrong when I thought it was a rock and it was a sponge. But look at the sort of the detritus in the bottom left right now.
Yeah, I wonder if
also
those
looks
are
like
also
sponge.
Yeah, possibly some skeletons that are left behind. Yeah, this looks like a Brisingate star. Very long. Yeah, it is a Brisingate starfish with these very long arms. And
is that
and we have a spot lobster as well at the base of it.
Yeah.
Yeah.
So have we seen any live sponges yet, or have we only seen
not yet. No, not yet,
sponge?
actually.
Yeah.
Oh, look at that. We also have a very small gastropod there to the left. But yes, this is clearly a sponge. And you can see, even just from the pattern on the surface, these water canals, which are part of the sponge aquifer system. And this sponge may actually belong to a group that has what we call the bath sponges. So unlike other sponges, they have a skeleton that is composed by a spongin fiber. So it's a very fibrous material, and not the spicules, the silica spicules as we see in other sponges. Look at that. The central plate of the Brisingate starfish. Yeah, very nice. And actually it looks like there's something else hanging on to it, like something purple, but I'm not sure what it is.
Oh, in the top left?
Yeah, and also here,
right there.
Some sort of worm, I guess.
Oh, hello, fish.
Look at that. Beautiful color. So it's interesting to see that any sort of substrate that, uplifts from the seafloor is immediately taken over.
Mm-hmm. And that's the closest they're getting to a hard substrate or one that's a little above the sediment. Oh, there goes another fish on the bottom.
We will do our best to get nice imagery of the fish that we're seeing here, so that our science team on shore can...
This is interesting. We're seeing a couple of little... Now we're finally seeing a few rocks.
Oh,
it's an isopod,
That is a huge isopod.
I do not like the isopods.
But that is a very, very large isopod,
yes, it
actually.
Is.
Yes, magnificent
Wow.
isopod.
I would not use the term magnificent. Frightening. These are not my favorites.
I have to say this is the largest isopod I've seen.
It's
so
very
far.
cool.
It is common that we see these large isopods in the deep sea. Wow.
Let's
Look
not
at
disturb
that.
it.
Sounds like Deb is very, very concerned that if this isopod moves, she will be more uncomfortable.
I will not be happy if this isopod moves.
Look at that.
That's a massive one.
I was trying to look at the rocks beyond it, but I don't really want to right now.
That must have been, 15 centimeters or 20 centimeters?
I would say 20 centimeters.
Wow.
Easily, yes.
It doesn't seem disturbed at all by us.
What could disturb that giant isopod?
Oh, jeez.
With that armor.
Right.
That's true.
So what, is this a crustacean?
Yes, it is.
Okay.
Yeah.
And what makes a giant isopod special?
It has this very strong exoskeleton there. Oh, look at that detail. Wow.
That's a face only a mother could love.
Oh. Don't talk about the mother isopod. It might show up.
But that zoom also gave us a little chance to look at the sediment. You can see how much biogenic sediment is there. You see some old shells. You see some... and then above it, we see some little outcrops.
I'm wondering if this could be in this genus Bathynomus. And there are almost 20 species known within this genus. So that's something we had not seen on this expedition before.
Yeah. I think we've seen them previously on prior expeditions with the Okeanos Explorer.
Yeah.
And every so often we're seeing actual rocks, many of which are sedimented, and they're very low relief. So we had seen a small outcrop while we were looking at the isopod.
So we'll hover here for just a moment while we handle a pilot change in the front row. In the meantime, we can reminisce about that isopod.
No. But did you know that, so Lang Bank is basically, it's a large carbonate platform, and it was formed mostly originally by the elkhorn coral, which I'm not going to try to pronounce its scientific name.
Acropora palmata.
Yep. And it was also, so that was the kind of the lowest part of the bank. And then it also has Orbicella favellata, which is another part of the reef building that has gone on below this bank. And then a lot of that, as it was getting cemented together and sort of forming the coral reef, there's also sand and carbonate rubble, and a lot of bioturbation over time. And so what we're seeing is just the surface of the slope that we're looking at, which is mostly sediment and probably a little bit of hard bottom. It's very interesting. because this bank could be over 9,000 years old.
Okay.
Could be even older. It seems like there might be some lively debate over how old this bank actually is.
So geologically speaking, that's actually rather young.
Yeah, that's all.
Yeah, that's a young.
Okay, our pilot change is almost complete. We'll be continuing to move upslope in the next couple minutes here. Our current depth is approximately 670 meters. So again, the fish range is for a lot of the commercial species that we've been asked to look for are something less than 600 meters. So we expect to see more dense and more diverse communities as we move upslope. But wow, that isopod, I can't get over that.
And the temperature here is about 8.5 degrees. I bet there's a lot of cool stuff if you dug just a couple of centimeters into the sediment, that you'd find a lot of interesting benthic organisms. All right, so we're rolling along.
Now this line of material that's moving across the screen here, this looks like bioturbation. This looks like a path, something left behind, evidence of something crawling through the sediment, probably looking for food.
Like an isopod?
Could be an isopod. I think the isopod we saw would have left a bigger track.
Probably. Is there a fish right there in the middle? I don't know what that is. Oh,
So
it is
for
a
those
fish.
watching at home, the lasers are approximately 10 centimeters apart.
And so is that fish. That's really cool. It's sitting on its fins. Go ahead now. We will, but not right now. So I think the idea is we're going to look for areas that have a little bit higher biodiversity, and that's when we'll probably fire off the Niskins. So, yep, no specific area right now that we expect to fire them in. It'll be more based on what we see. It is this fish is very cool. I don't know what it is.
I'm not sure this is what are usually called these tripod fish. So they use these sort of long barbels and the pectoral fins to stay up above the sediment. Oh, it's really cool.
It is very, very cool.
Yeah, that's probably what it is, these structures coming off from the top of its head, serving as sensorial to either detect possibly prey.
And they look like flanges when we were looking at them just a little bit ago. They look like they widen at the tips.
Yeah, at the tip. Yeah. Oh, yeah,
like
look at that.
Feathery structure at the tips. Now might be a good time to say that there aren't any fish experts in the chat room with us right now.
Yes, that is true.
Otherwise, we'd probably have a great identification of this very cool fish. But I like your tripod fish idea. It certainly seems quite reasonable. Oh, it is a tripod fish. Bioturbation bigelowii? Boy, I'm sure I did not get that right.
So in the family Hypnoidea, Bioturbation bigelowii. Yeah. So we got a tentative identification here. And for a moment I had a feeling there was something large going over us there. Yeah. Yeah,
yep.
It looks like the same fish.
This one's kind of posing. Opening its mouth.
He seems to have turned into the current. I don't know if that's a little bit of backwash from the ROV or current that we're actually experiencing, but
What's on the top left that's bluish looking?
Yeah, something purplish there. Is it also a fish or?
anytime there's a purple lump on the seafloor, I get excited for a holothurian. But I don't know.
Do you like holothurians?
Yeah. Yeah, and it really looks like it's an holothurian here.
Yep.
Rather transparent.
It's interesting to me how many of these species or these organisms are not disturbed by us. We've had a lot that are just like, nope, going to keep hanging out.
Yeah.
Yeah, it does look like a holothurian, doesn't it?
Yeah, it does.
Oh, I like the spines, though.
Yeah. And so holothurians are detritivores, meaning that they will, eat the sediment and then take from the sediment any organic matter that there might be, then returning the mineral part of the sediment to the seafloor.
And they have to really eat a lot of sediment.
Yes, a lot to take some organic matter from it, yeah.
Thanks, pilots. That was great. This is beautiful. Yep, so it's about over 10 centimeters big. So we're not seeing a ton of biodiversity yet, but we have seen some black corals already, and we've seen a number of fish and a bunch of dead sponges.
Yeah.
And the isopod, which we can't forget.
The giant isopod.
Hopefully we will not see another one of.
You will have nightmares tonight.
Yeah, they are not my favorite. I'm sure they serve a very important role in the deep sea food chain, but wouldn't want to come face to face with one.
Oh, there's probably just a bit of sargassum that has come down from the surface. This is what's been keeping us
yeah, we've
out of the water.
Yeah, the last 2 days we have not been able to dive because of the amount of sargassum, not just at the surface, but about 15 feet down. It's been wreaking havoc with the ship because the ship uses seawater to cool its engines, and it pulls that water in just from the ocean, and it's been pulling in, unfortunately, sargassum as well. And that's been causing strain on the engines.
Really clogging the system. And
what do we hear? Is it record sargassum time right now?
Yeah, apparently. Yeah. We've chose the right time to be here. Mm-hmm. And here's another, seems to be a rather large dead sponge as well.
You think if we keep going upslope, we're going to see live sponges?
I'm really hoping so. Because we've been seeing so many dead ones, so... Yeah, this one is also quite large.
That is a big sponge.
Yeah, it is. Look at that.
I'm surprised there's not something colonizing it.
Oh, there's a shrimp underneath it, using it as a shelter. Mm-hmm. Yeah, but you can clearly see here those water canals. And you can see how busy the shrimp is here feeding. You can see those buccal parts just,
nomming.
Yeah. Oh, and there's a tiny little hermit crab as well to the right there. And so these type of sponges, the type that traditionally were called keratose sponges, so these are the ones that are actually quite squishy, because their skeleton is built by this spongine fiber, so this very dense network of a fibrous material, which then soaks up. You can see if you put it in the water, it will soak up a lot of water.
This whole seafloor is just really heavily bioturbated.
It is. Oh, another one of these tripod fish.
Guess we're going to be seeing a lot of these today.
In the family Hypnopidae. Yeah, we have there another Olathurian. looks somewhat different from the previous one.
It does.
Previous one had a more bluish sort of coloration.
And it was more blobby.
Yeah,
this one's much more cylindrical.
Yeah, that's true.
Yeah, and its spine, I think its spine system is different.
It's rolling over. Oh, let's have a look at that. It's rolling over for some reason. Oh, and you can see there the mouth.
Mm-hmm.
Look at that. Oh.
That's a nice view.
Oh, interesting. And now it stopped.
Maybe it wanted to go to a slightly different part of the sediment for breakfast.
Probably.
Just needed a good morning stretch.
Yeah. Roll over.
A good morning roll.
Yep.
So this is dive 7 of our Voyage to the Ridge expedition. This is our third dive in and around Puerto Rico and the U.S. Virgin Islands. It's been very interesting so far because we're seeing a lot of fish diversity. We're seeing some holothurians. We're seeing these dead sponges. But I expect that this is going to get even more exciting as we move up the slope. Mm-hmm. And we're here on Lang Bank, which is about 9.5 miles northeast of St. Croix. It's sort of the eastward extension of St. Croix. And it is a very important fishery spawning ground.
Oh, look at this. Look at the eyes.
That is really big eyes.
Amazing. Yeah. Wow.
That's very cool.
It's like a rainbow. Look at that.
I've never seen eyes that look like this on a shrimp.
Yeah. Amazing.
Wow.
Yeah. Beautiful.
And these cameras are just amazing
Yes, they are.
on the ROV. So they have a 18 times zoom, which is why we can routinely look at things that are, less than, 2 or 3 centimeters on the seafloor. We can
such
really,
detail.
Yep. So unfortunately, we kicked up a little bit of dust right now, so it's a little obscuring our beautiful shrimp eyes. But it is really amazing how close and how detailed we can get.
Oh, there
Run!
you go. Stay, please stay.
Run!
No. Wow, look at those eyes.
And one of our scientists in the chat room, Nolan Barrett, is saying that the eyes remind him of the shallow water mantis shrimp, but this is actually really definitely a shrimp, a true shrimp.
Wow.
And it's really cool. And so our pilots are saying that about 20 meters ahead there is a target. So there maybe there's a small outcrop. We'll find out. But as we move there, we'll continue to look at everything on the seafloor. The seafloor does have a little bit more relief, just a little bit more than when we started, seeing what looked like little mounds here and there.
And I think we're seeing also some urchin tests, yeah, spread out on the seafloor.
Yep, there's a little bit of a carbonate hard bottom to look at. Just a small outcrop. Interestingly, there's not a whole lot on it. At least.
But also with the sedimentation, it's not uncommon, yeah.
And there is a lot of sedimentation here right now.
So typically a lot of, sponge and coral species look for sort of, let's say, clean, hard substrate that can be used for, to get a really good attachment point for them to grow on.
Oh, interesting. I don't know.
Those look like trace marks of some sort, but...
Oh,
that
wow.
is very interesting, yeah.
We just put it on in the back so we could look at it more closely.
So for those at home, the pilot drew our attention to the texture and the sediment in front of D2, which you can see very clearly on stream 2 if you switch over to the Sirius view.
I have
looks
to
like
assume
these trails.
Yeah, that has to have been biogenic.
Biogenic, so some organism that is...
That's really interesting.
Yeah, and it's interesting because when you see just from the Deep Discoverer camera, you don't quite realize the pattern that is creating, right?
I would highly recommend everyone move to camera 2 right now, just to take a look at it from on shore. It definitely has to be biogenic.
Yeah.
The dance
so
of the
possibly,
isopods.
Yeah. Oh, could it?
Or the holothurians.
I wonder if it could actually be one of those isopods creating this, because looks like it's a relatively large and wide trace mark as well.
And it curves around. Are there any ways to identify what creates that based on the shape? Like, do holothurians like to move in straight lines? Or
I think it's very difficult. So ideally, we would see the organism that is causing it at one of the. But this could definitely be an echinoderm.
Oh, it sounds like Chris Ma has popped on.
Hi, Chris.
Hi. So I'm looking at the view right now, and to the left, there is a little bun-shaped thing that is sitting right in the middle of the trail.
Yeah.
Do you see that?
Yes, we do.
Because that looks like the urchin that's causing the tracks that you're looking at.
Oh, that's very interesting, Chris.
So if we could switch over or zoom over or zoom on that. Because I think this is one of the coolest. I will hold off on the name until I see it, but I believe it's an urchin, and I think I know which one it is, but I want to make sure we can verify that it's actually an urchin and it's not a rock, and I'm not going to embarrass myself.
I can tell you a rock probably didn't make that.
Like a little hole in the middle.
So let's have a look.
Yeah.
If we can.
So pilots, can we zoom in on the sort of lower center?
Yeah.
I think that's what Chris was talking about, right?
Yeah, I think so.
Yeah, and there's almost a black hole, a little black dot in the center of it.
Yes, it is.
And hopefully I didn't just identify a rock.
That is not a rock.
And I don't think it's a sponge either.
Oh, it is. It is an urchin, yes. Okay, so I'm not sure if it's alive, but this is probably what had caused those tracks. This is the the skeleton of a sea urchin called Conolampus. And it's an irregular urchin, which is almost pleasingly shaped like a bun. I mean, it literally. I have skeletons. We have material of this in the collections. So this is a sea urchin, and it's what's called an irregular urchin because they're modified to digging through sediment rather than just sort of sitting casually on the surface. And so all of their spines are modified into either very short kind of hair-like structures, and a lot of them are modified into helping them move and dig through sediment. And this particular one, Conolampus, is particularly well known because it makes these little tracks along the bottom. It kind of, if you remember, I don't know if you remember these, there were these TV commercials called Scrubble Bubbles years ago, and they look just like that. They look like Pac-Man ghosts. They're kind of very kind of bun-shaped Pac-Man ghost, Mr. Bubble-shaped things, and they just make these delightfully little like linear, or not linear, but they make these very distinct trails in the sediment. And what it looks like is, I mean, unfortunately, it looks like it's dead.
Yeah.
But I suspect what happened was, for whatever reason, it moved around and made all of these tracks. And these tracks are probably, they could be anywhere from months to years old.
Wow.
But that skeleton, looks dead because it has a lot of fouling on it. But there could be others in this area. I don't think they dig per se, but if we come upon more kind of mound-like structures, I mean, they will probably be difficult to tell until you get a recognition, an eye view for them. But they do occur in this area, and they will create more of these tracks. So let's keep an eye open for them. And and that was unfortunately a dead one, but like I said, exciting to see one of those. They used to see them frequently with the Johnson Sea Link. So
brilliant.
Anyway, I'll let you guys get back to it. Looks like you.
Brilliant.
You got a
Thank
fish,
you.
so.
Thank you, Chris.
Very cool. Thank you, Chris.
We'll keep an eye out to see if we find
Oh.
one of those alive. We wouldn't actually have seen.
I hope so. Thank you. Bye-bye.
Thank you. We wouldn't actually have seen this if it wasn't for the serious view.
Thanks so much. Bye-bye.
Bye, Chris. So Chris is one of our echinoderm experts.
Yeah.
Working at the Smithsonian.
Yeah.
And always calls in if he can when something exciting echinodermish comes up on the screen. And that was very interesting because, as he was saying that was a dead sea urchin, and yet the traces were still on the seafloor. And that sort of correlates with not a very high current here, that those were able to stay without being bioturbated upon, basically, or having a current come and sweep them away. It's another black coral.
Another of these Stichopathus black coral here. And we see again there to the top right a test of another urchin. Not sure if the same species or a different one, but we've been seeing a few of these actually.
And after they're done zooming in here, we see a little bit of a difference in the sediment where this black coral is versus just upslope. So what is this attached to on the seafloor?
Let's have a look at the holdfast to see if we can see exactly what it's attaching onto. But looks like some sort of a small rock, a small pebble. We see also to the right a small sponge. But so there is some of this hard substrate here and there.
So it found one tiny rock.
Exactly. Yeah, this looks very similar to the other Stichopathus corals we've been seeing throughout this dive.
Just the water column is full of life here. Things keep
flowing by.
Yeah, quickly.
Is that a can or some sort of metal there?
It
Also
does
to
look
the
like
left,
metal, yes.
right? Yeah.
But we're looking at this circular object that's coming into the center view that might be an urchin.
That's another urchin test.
And just look at the difference between where we are right now,
Yeah,
the sediment,
and to the left.
Yep.
Yeah.
And so it just, the character of the sediment has changed, and it's fairly hard. You know, one side looks completely different than the other, not a lot of transition.
Yeah, and it seems actually to be the same one that Chris was talking about. So
Or something similar, yeah.
Yeah, something similar, Conolampas.
Is this
but
one
again,
alive? It does not look alive.
It doesn't look alive, no.
Yep.
So you can see the skeleton is like pretty cleared of any spines. We don't see any of those tube feet structures. Yeah, and it's
You can
it's
see the
covered
holes in the
already.
chest.
Yeah,
That's really interesting.
Yeah, and it has already a lot of sediment and debris over it, and even some serpulid worms. You see those calcareous tubes there.
But I thought what Chris was saying about that last one, and I, of course, could be completely wrong, is that most of its spines were actually underneath the test that we're seeing. That they would be on the bottom, and you wouldn't necessarily see them.
I'm not sure, but I think we would still see them along the top surface of the test.
Oh, look, we found a rock.
Oh, we found a large rock, actually. Yeah. Look at that.
We
very
reached an outcrop.
heavily sedimented still.
Very heavily sedimented, yes.
But already here you can start seeing a bit more life. I'm seeing there's those...
Could we zoom in on the bottom left?
Yeah. Let's have a look at that, because I bet those are sponges. It's difficult to say still from a distance, but they do, some of them do look like. Wow. Yeah, and Chris here in the chat room is saying that, yeah, probably they would be on the surface as well, but very short. Oh.
What are these?
These are... Actually, these are not sponges.
Yeah,
no,
they look like oysters.
Yeah, they look like oysters.
What are they?
And we have a look, and they are oysters of some sort. Look at that.
Okay.
Yeah. Well, we also see a few...
Are those tube worms to the right?
Some tube worms, and we see also a few small sponges and what seem to be either some bryozoans or stillasterid corals, these very small white branching structures, but the large white or pearlish-like structures are oysters, in fact. Oh.
Do you think those are all oysters, or do you think that
no, I think
some of them might be... Well, that one's open on the bottom.
Yeah, that one.
But, I mean, could it be a scallop instead of an oyster? I don't really know much about these and how they would look different.
Yeah, that's a good question. Some of them actually seem to just have one of the valves, so probably dead already and just open.
But the one that
they are. Many of them,
in
fact.
But we've seen two that had both of its shells, and you could see the tissue between it.
And you can see how some of the serpulid tubes there are already cemented. They look like cemented and integrated into the rock. So this is like a biogenic rock, really.
Oh, yeah.
An aggregation of all sorts of different materials there. We do see some really small sponges here and there.
And a shrimp.
And a shrimp there. Making use of one of those crevices in the rock.
But yeah, I mean, this this piece of rock looks like it's probably a limestone that was created with all the different biogenic materials. And many of the rocks around here are a combination of old corals, sand, carbonate sediment, and then as we see here, void space. You see holes in a lot of these rocks. Very cool.
So you think this would have been formed, this is in place, or it would have come from a higher area of the bank?
I honestly don't know. I would probably guess that I think we'd have to kind of keep seeing it, but if it's a block by itself, I would guess that it came from somewhere above.
We can see from the serious view. It's
There's
a relatively large
big
block. Yeah. And we can see that some of these tube worms or serpulids have been probably here for a long time, because many of them have already also some coating and really fully integrated in the rock. Is this a crinoid also coming into view there? Hanging on that rock.
Little squat lobsters to the bottom right.
Oh, there it is.
I don't know why I find those cute, but I do not like the isopods.
Yeah, they look like, the isopod looks like a Darth Vader, isn't it?
Not a fan of the isopods, I should say. We just tried to track that fish. It's wandering out of screen right now. Oh, what's the pink thing on the top right?
There's another black coral there. Possibly the genus Bathypathus.
Yeah.
Yes, please. Yeah.
There's a lot going on here.
Yeah, there's actually quite a number of different things here. But let's perhaps have a look at the black coral, or we can have a look at the sponge as well.
So are these pinkish orange things off to the left are the other Stichopathus?
Yeah, those are the
we've seen before.
Stichopathus black corals that we've seen earlier on.
Those are the ones that have kind of a spirally
coil,
shape.
Yeah.
Yep.
There's actually another one there to the right as well, but the one to the left now, it's a different species of black coral.
Yep. And is that a crinoid on the top right?
Yes, and that one is a crinoid. So the one to the left is probably in the genus Bathypathus. It has this sort of central axis, and then you have this secondary axis coming on from that center there. But we would need a better zoom to see. Up in the Mid-Atlantic Ridge and Azores Plateau, where we were earlier, we've seen also some of these Bathypathus, possibly the species Bathypathus pseudo-alternata. But I'm not sure this could be the same species.
Is that a shrimp right behind it?
Yeah. So let's see if we can have a better zoom of this one. And you can see here, it seems like there is a bit more current. So they also take advantage of this current to get food particles.
So, Joanna, it looks like all the polyps are pretty much on one side of
that's true.
Of the node. No, not node. What's the word?
Yeah, that branch.
The branch.
Yeah.
Yep. So the pilot is asking what
I think those are the part of the polyp itself.
Yeah, the pilot is asking what the pink pieces were, the little pink bulbs between the polyps.
Those are part of the polyp itself, and
we're waiting from the chat room from our onshore scientists to give us a real identification. Yep, Bathypathus, Pseudo-alternata.
Oh, Pseudo-alternata. So it would be the same species that we actually saw in the Mid-Atlantic region, the Azores Plateau. So that's very interesting.
But look at how sedimented this outcrop is. Boy, there's just, it's coated. Yep, there's another little black coral, which is probably Stichopathus on the bottom left, and there's a brittle star right next to it. Yep, so that's what looks like probably two Stichopathes with a...
smaller ones and a brittle star.
Mm-hmm.
And this brittle star...
And boy, this rock is really covered in sediment and covered in biology. I wonder what the little white thing behind the brittle star is. Is that possibly a hydrozoa? Ooh, ultra zoom. Well, this gives us a great view to look at. And going back to the pilot's question earlier about what the pink bulbs were between the polyps on that Bathypathus, evidently those are mouths. Is that another little hermit crab? I think it is. It's a hermit crab with some kind of test to the right. But it looks like it might have taken an oyster shell or part of an oyster shell. I don't know. It's thriving in its home. And this really just shows how important rocky outcrops are, hard bottom areas on these banks, because we're seeing so much more diversity and so many more animals just on this one outcrop than we did for pretty much the whole beginning of this dive. Even though it's still fairly sedimented, they're finding this to be a more hospitable place to live than the sediment that we saw a little further downslope. So I think one of the questions is whether this is just an isolated block that happens to be sitting in a pile of sediment or whether it is part of a larger outcrop. From the Sirius view, which is camera 2, you can see that we're looking at a giant boulder. That boulder is bigger than the Deep Discoverer ROV, which is pretty big.
Yeah, let's have a look if this is the same Bathypathus that we've seen earlier on. Yeah, we can see there's a bit more current here, isn't it?
has one big rock.
Yeah, huge block.
Mm-hmm.
And we're here now at 654 meters depth.
Mm-hmm.
We are hoping to get as shallow as 300 meters towards the end of this transect.
And we are hoping that once we sort of cross the 600-meter boundary up into the 500-meter boundary, that's when we're probably, hopefully, going to start seeing some of these commercially important species of fish.
Like the queen snapper.
And we have a lot of time on this dive. We have many hours. Gives us time to check things out. What is off to the left?
It actually looks a little bit different, this this coral.
I think they're having a hard time holding the ROV steady in this particular orientation.
Yeah.
So what is that off to the left behind it?
It's difficult to see.
Yeah.
This actually looks a little bit different. I wonder if it's the same Pseudo alternata that we've just seen earlier on. It actually looks the secondary branches are coming out from that central stem at different heights. So alternating between the right and the left. And the other ones seem to be more coming from the same central point. But
one of our scientists thinks it is the same species that we just saw.
Okay.
And they're the coral experts, so we listen to them.
Yeah, we sure do.
We are very lucky to have a robust group of shoreside scientists who are willing to join us on dives and help to identify the species that we're looking at, because a lot of them look very similar to one another.
Yeah, that's true.
And so it's interesting. Sometimes you have to zoom in on specific parts of a coral.
We have again this crinoid that we found on the previous dive, the Holopus.
The one that looks like a fist.
Yeah.
Awesome.
That Chris was really excited about. And so this is an holopodid crinoid. And there's something about them that make them look like prehistoric, and indeed it seems that they have been around since the Jurassic, so many million years ago. And instead of having a proper stalk as other crinoids, these actually cement directly to the substrate. And as you can see now, these arms, which are relatively short, right now they're fully closed, and they make this sort of like a
very tightly closed fist.
Yeah, exactly.
Yeah. And on one of our previous dives, we did see several of these, and one of them was more open. You could see its arms
with
extending
the arms
out.
Exactly. And so it seems that in this family there are only 6 species, 3 of them in the genus Holopus and then 3 in the genus Cyathidium. And we've seen Cyathidium in the Mid-Atlantic ridge on the Azores Plateau. But here around Puerto Rico, we've seen just this one, Holopus.
But evidently these are fairly common around the waters of Puerto Rico, and so it's probably not surprising that they're also here in the waters off of St. Croix. In 2018, when the Okeanos explorer was visiting Puerto Rico, they actually encountered this a number of times. And so now we're moving across the top of this very large rock. Oh, there's a tube anemone, I think. I could be wrong.
I think we had someone calling in.
Did someone join the conference line?
Hi, this is Michelle checking in.
Hi, Michelle. I think
so
that's
we're
a
zooming
fly.
in on a
That
crab.
looks like a fly trap anemone.
Yeah. Oh, a fish going by?
Uh-huh. Oh, this is one of those carrier crabs. Look at that. So it has these modified legs to hold some sort of either, a sponge or a coral. And we think that they use it for as some sort of camouflage as they travel around the seafloor. Look at that, and I think it's holding a bit of a sponge. In
But
this
dead,
case,
right?
yeah.
Yeah.
It's,
Or not?
it doesn't look completely dead to me.
It doesn't look completely alive to me. That's amazing.
Yeah.
You know, John, next time we see this much abundance in one place, maybe we should consider taking a water bottle. We kind of are over the top of it now, so perhaps not a good time to take it. But
See. Yeah, let's see
No, I just like pushing buttons.
a bit further up what we can find.
Yep. For those who are on shore, we have 4 water bottles left in our dive. We took one in the deep scattering layer. And these are for eDNA, and the idea is to look at the community assemblages. So you get, you don't get specific DNA about one particular thing, but you learn what
Of
the whole
entire
community.
Yeah. Yeah, this is a fly trap anemone. Because it reminds a bit of those carnivorous
Oh yeah, it absolutely
plants.
looks like that.
Right.
So that's different.
And
we
they will
haven't-
work, and they will actually work quite similarly.
We didn't
So
see
they
one of
will
these
close.
yet.
No,
right?
no, we didn't. So this would be in the family Ormathidae.
Yeah, it really does look like
Yeah.
a Venus flytrap.
And we can see the mouth there at the center.
This one's beautiful. Oh yeah, you really can see its mouth.
Yeah, it is very clear there. It's really nice tentacles all around this central disc, and when some prey will come by, it can actually close these sort of flaps over it. Great shot. Yeah, and we can see here how how well attached to the substrate it is. This base, this very broad base. Great image, thank you.
And so we saw that one large rocky outcrop, and now we're sort of back into the sedimented, very bioturbated seafloor. And it's probably a combination of carbonate sediment, biogenic sediment. So as things die and disassemble, they end up here on the seafloor. I'm really interested by these little mounds, though. What's creating these little mounds?
Yeah, I think those could be some of those quad lobsters that we've seen.
Oh, really?
Yeah.
They create mounds?
We could, yeah, they do. Sometimes they have these burrows and they can make some mounds. But it's hard to tell.
Yeah. What's coming up here on the right side as we move up? See lots of little white things.
Yeah, I don't know. I wonder if some pieces of coral or...
I don't know.
Looks quite biogenic.
And then there's something very, very dark right in front of us.
Yeah, what is that?
So I don't know, this whole area is very interesting. Pilot, if you have a moment, can we do a snap zoom on the sediment in front of the dark thing and then on the dark thing? I feel like every time I say, is it a fish, it's not a fish, so I'm not going to call it a fish until we know it's a fish. Maybe it's a holothurian.
It looks like a bottle there. Yeah. Oh, yeah, and this looks like coral rubble.
Yeah, it is very much coral rubble.
Yeah.
What's the pink thing in the bottom right?
Probably some sort of shell or... Yeah, there's actually a few shells here as well, yeah. Different types.
Yeah. And definitely some coral.
Yeah.
Very cool.
And several fragments here of solitary corals, scleractinian corals.
Are there cup corals too here?
Yeah, that's the one we saw. We saw one just now.
Okay. Should we bet on whether that dark thing was a holothurian?
I think it was a bottle.
You think it was a bottle?
I think so, but I'm not sure.
Hope springs eternal. I'm gonna keep hoping it's a holothurian.
And it's a bottle.
Man. Definitely a bottle. Not a holothurian.
No. And they're stalked of probably a black coral. Tripod fish there at the back.
Yep.
Oh, a Stichopathus coral here to the right. And there seems to be something on it as well. So perhaps we could have a quick look. Yeah. Oh, it's again the brittle star. Oh, interesting. So in the previous dives that we've seen the Stichopathus corals live, we didn't see the brittle stars associated to them. So this must be like an unusual association that's occurring here.
I don't know if it's unusual or if we just didn't see it
earlier. Yeah, we probably just didn't see it earlier.
I mean, but it seems like a very reasonable thing to do because it gets them higher up into the water column and allows them to do some interesting filter feeding at a higher level.
That's interesting. Yeah.
They are actively moving right now. It's very cool.
Yeah. Look at that, really tightly coiled around it. I'm not sure how common this is. Also in the Mid-Atlantic region, the Azores, where we were previously, when we'd see these coral, these Stichopathus corals, we never saw this brittle star associated with it.
Is there any chance we could zoom a little bit more closely on the central disc?
Of the brittle star, yeah.
So we're waiting for our Echinoderm expert to tell us what kind of brittle star this might be. I think that's probably good enough. What do you think? There's definitely a little bit more hard bottom here. There's some little outcrops of rocks from which we're seeing the Stichopathus on. So that's an Astrogomphus. Oh, I'm really not doing well.
Astrogomphus.
Their spines on the disc.
This brittle star. Okay. we'll be able to actually have a closer look here at the central disk of the brittle star.
Thank you so much for the beautiful imagery. This is fantastic. So we're going to lose Joanna in just a couple minutes. She's going to go talk to some children in the Azores and tell them about what we've been seeing on our expedition.
Yeah, so we'll have now a live interaction with Expo Lab at the island of San Miguel in the Azores, where we will share a bit of our findings when we were in the Azores Plateau in the Mid-Atlantic Ridge. But then I will be back in half an hour or so.
I am really hoping they send in a biologist to help me for this. But thank you so much. Have a wonderful time.
Thank you.
Pilot, this is Watch Lead. One of the shoreside scientists is asking whether we're seeing more current than we were previously. Okay, thank you. Okay, great. All right, so we're going to keep moving. The current hasn't really changed since we have started our dive. It's coming from east to west and fairly minimal. And the temperature is about 8.7 degrees Celsius. We are at a depth of 648 meters here at Lang Bank, which is northeast of St. Croix. Lots of cool stuff to see. It's a little few and far between, but still pretty interesting. You think that's a sea urchin? We'll find out. So we've seen a number of these tripod fish, which are very cool. They're just kind of using their fins to get off the bottom so that they can get a little bit more food flowing by. Is it an urchin? Yeah, so that is a sea urchin. It does look like it's kind of on its side, and it's really nice. You can see the different spines. Some are straight and pointy, and some have a little bit more of a bulb shape. They look like a Q-tip. And it is actively trying to move, I think, perhaps because of us. So this is an Echinoderms. I wonder what the difference is between the spines that are sort of more bulbous at the end and those that are finer and thinner. And then there's another type too that we see on the left that looks more like a golf tee, a very small golf tee. And our Echinoderm expert, Chris Ma, is going to join us in just a minute to probably tell me about all the things that I've just mentioned that are incorrect about this beautiful Echinoderms. So hopefully we can stay here and look at it a little bit longer.
Hello.
Hello, Chris.
Hi, Deb. So first, I'm just going to say the new automation on the phone takes forever. So this is really interesting. I always like to call in and talk about these. These are so the scientific word I was using, the Echinotheridae. The Echinotheridae is not necessarily the exact family, but just the Echinotheridae group. And the reason I'm calling it that, these are what's called pancake urchins, or sometimes they're called tam o'shanter urchins, because basically when these are removed from the water, the entire skeleton of these animals is really soft. And when they are removed from water, the pressure is what basically suspends the animal's shape, and they collapse into sort of a flat pancake shape. One of the characteristics that I can use to identify them is the fact that from around the surface of, so this one's weird because it's actually upside down.
Oh.
What we're seeing, like right in the center there, that's the mouth. That's the Aristotle's lantern. And around the edge of the urchin, you'll see those horseshoe-shaped spines. Some of them look kind of like the tip of a cane or something. So those are characteristic of this group. And they literally use those to walk around on. I mean, they use all their spines in locomotion, but they have these particular spines, which facilitates their ability to walk around on sediment. We're seeing a bunch of things. The long tube feet, which are extended beyond the spines. We're also seeing a lot of these interesting spines with the club-shaped bits on them. Those are probably things that facilitate its defense, because in the shallows, these urchins actually have representative, their relatives, and they're called fire urchins. And the deep water urchins of this group also contain venom in the spines. And that's one of the reasons why I tend not to, I don't know urchins, but I don't ever ask for them to be collected, because collecting them usually entails some risk to whoever ends up having to deal with them.
Oh, my.
But those bags that are on the spines, I'm sorry?
Oh, that's fascinating, Chris. And
I'm sorry, what was that?
Thank you for not asking us to collect things that have venom in them.
Yeah, but those little puffy bags at the end are probably the equivalent of sort of the bags for hypodermic. Oh, there's a Echinolepis. The puffy bags for a hypodermic needle. So you could, at least that's the speculation, is that these are all defensive structures. So those little round bits that we saw are similar to the big, I don't know if you remember the Formosoma placenta urchins that we saw. This is the same kind of urchin. And the tips, you remember the surface of those urchins have those big bags? That's the same. Those little round club-shaped things are the same thing. They're tissue around the spines. And so they're probably, intended for defense against fish or some other type of thing. But they're, the documentation for the deep water ones is not as well known as the shallow water ones. But it's usually, I don't know if it's actually even published that the deep water ones can sting you. So, it depends on species and size of the urchin, from what I'm told. But they're quite sharp. But they're really interesting animals for a number of reasons. The locomotion, the occurrence. They were actually a group of urchins known from the fossil record before they were discovered from living specimens. Here's a nice Citrus blakei urchin. So the lower end of the screen, the spine. This is an urchin with the spines that look like big, flat flanges. This is one of these weird. In other places, we would call them pencil urchins, but oh wait, am I getting it wrong from the distance? Okay, no, it is just a bouncy. Okay, yeah. So I'm sorry, it's not Citrus blakei, but it is a Citroid urchin, and so so Citroid urchins are also are all the same kind of urchin, like the ones we saw the other day with the big flanges on the spines, but they don't have tissue on the spines. And so, there's a nice little squat lobster living there. And so one of the things you'll notice is that there's actually fouling on the spines. There are serpulids, there's bryozoans, there's all sorts of little, and certain urchins actually have specific types of fouling organisms which they prefer to carry around. It's not entirely certain how or what the dynamic is. If the urchins, carry them around as camouflage, as against, smell of other predators, or if there's just some other benefit that they gain from them. But the interesting thing, too, about Citroids and Echinotherids is that they both have this historical component. Citroids are known from urchins in the Paleozoic, and those other urchins that we just saw are also in the fossil record. So there's almost a very archaic nature to them. They're not obviously like fossil species, but they're very similar to species in the fossil record. And they are probably very ecologically important because urchins like this Citroid urchin, people often attribute urchins to being just herbivorous and generalist animals. But in fact, Citroids are predators, and predators that feed on crinoids, the stocked crinoids that we see elsewhere. And they will actually attack them and chase them down. And granted, that's a very slow chase, but it is something that we've seen on Okeanos and something that has been seen by other scientists on research dives. It's very interesting because they think that's possibly one of the reasons why feather stars have evolved swimming behavior, and possibly even why some urchins, or why some feather stars have evolved the feather, like why there are, so there within crinoids there are both stalked crinoids and feather stars, the ones without a stalk. And so some people think that the feather star morphology might be an adaptation to the stalked to surviving predation by these urchins. So there's a whole bunch of echinoderm evolution that's tied up with understanding Citroids, and so they're very interesting for any number of reasons. And learning about what they do in the where they live is part of this whole puzzle, and a lot of it is still being learned. So anyway, we're not looking at yet another urchin, are we?
No, we're not. I don't think so, at least.
Sea anemone. Okay.
Yeah, but actually, Chris, I have a question for you.
No, it's sea anemone.
Yep.
Yeah.
When the last Citroid
please.
urchin that we were looking at, the central spines were like a sort of a dark pink, while all the others were much lighter, white. Is there a reason for that?
Yes. I don't know. I mean, other than the possible significance of the fact that red shows up as black in this environment. Mm-hmm. I mean, to be honest, I know that coloration is probably useful in diagnosing them. From the human perspective, it helps us figure out which one it is, and I need to go look it up. In my handbook of tropical Atlantic Citroid urchins. Mm-hmm. But like as to the functionality, I'm not certain. It could simply be incidental because Citroid urchins are generally colorful. But yes, it is very noticeable, and I wish I could give you a better answer. But like those Echinotherid urchins, most Echinotherid urchins in the deep sea are pretty drab colored. But when you get into the shallows, they're brilliantly. They're called fire urchins because they are bright orange and red and and blue and. And that's basically nature's way of saying do not touch. But down here, it's it's all quite sedate compared to things that are in the tropical reef setting. So, again, all sorts of mysteries that we've yet to understand. So, anyway, thanks so much for obliging me, and I hope we'll get to see some more spiny things in the near future.
Thanks, Chris. Always great to have you call in.
Oh, thanks. Well, the more you focus on echinoderms, the more I'll call in. Thank you. Bye-bye.
So that was fantastic. We just learned a lot about several urchins that we've seen this morning. And we did get to fly over a couple of different fish. We saw a holothurian as we were moving. And we're still just continuing our way upslope here at Lang Bank. We are at 637 meters. What do we think? Dead sponge, old rock, something on it?
It's kind of interesting how often we get sponge or rock.
Yeah.
Until we take a close look at it.
And a very big shell.
Yeah.
Like that's a gastropod shell, right? That looks like a sponge to me, personally.
Yep. Yeah, I agree with sponge.
And has a tube anemone at the bottom. And
maybe one on the other side. Yeah.
Yeah. And look at that. It just these sponge habitats that are no longer living really provide a place for a lot of other animals to live on it. That's a serpulid worm at the bottom, I believe. Oh, and there's a tiny little something. And I think we might be at the limits of our zoom right now.
Could be a lobster. Yep.
I'm not going to speculate. It's a little too small for me to see.
Yeah.
But it's really interesting. You know, there's just so much stuff sitting right on this one sponge. Provides a habitat for so many animals here. And for those of you who have been on the dive for the last hour or so, as you know, what we're doing is trying to move up the slope, quietly and slowly, with the idea that when we hit around 600 meters or even possibly 500 meters, we might see some commercially important fishery species. This area is really important for spawning of fish. That's a big shell. It'd be great if we could see inside it. Unfortunately, it's oriented not the right way for us to be able to sneak a peek in. I feel like I may have just accidentally issued a challenge to our pilots. That was not meant to be a challenge. You don't have to look in the shell. And there are a lot of these little burrows on the seafloor, little mounds, and they might be shrimp burrows. And the snail shell was identified as either a conch or a whelk snail shell. And we have been seeing a lot of these very small burrows on the seafloor as we've been moving up. Actually, it's pretty much since we got on the seafloor, we've been seeing them. We're just doing a quick change with the ROV pilots in the front row.
We'll have some short introductions here for the folks in the front row and the back row. We do change regularly as we swap pilots out to make sure everyone is fresh.
All right, I'll go ahead and introduce myself first. This is Jonathan Mefford, sitting pilot. I'm with the Global Foundation for Ocean Exploration, a group comprised of video, data, and mechanical and electrical engineers and various backgrounds. And I just swapped positions with my co-pilot here. I'll let him introduce himself.
Sitting co-pilot, this is Todd Gregory.
mechanical engineer with the Global Foundation for Ocean Exploration.
And to my left?
And sitting nav is Mark Durbin, network engineer with the Global Foundation for Ocean Exploration.
And I'm Brian Doros, broadcast systems engineer for the Global Foundation for Ocean Exploration, sitting in at video and broadcast control.
And this is Anna Sagatov. I'm sitting in at the video clipping station, and I'm also with the Global Foundation for Ocean Exploration.
Anna, can you tell us a little bit about what a video clipping station is and what you're doing there?
Sure. So I am sitting at a station where I can see we're currently recording 5 different recorders at the same time, including, so the D2 cam and Sirius cam are recorded consistently throughout the dive. And then recorders 1, 2, and 3 we can switch out, but usually they're on 3 of the other cameras that are on D2. So if we need to see another angle of an organism for collection, for example, we can switch to one of those recorders. And so I choose which camera angle is the most important to record. And when we see an interesting organism or a scientist has some important information to share about what we're seeing, I'll clip out that specific piece of video and I send it through our system, and that goes out to, we not only make videos with that footage that go to YouTube and go through NOAA's distribution, but we also send out to scientists, and they are able to see everything that we've seen during the dive. So that's what I do.
That's great. And actually, every day you also make a highlights video, right, of something that's very intriguing or interesting about each of the dives.
Yes. So we take turns. We've got a team of 3 video editors on this cruise, and so we kind of switch between who's doing which job. And today it's my day to make a highlight video. So I will basically just make a decision about what seems like an important or interesting thing that we saw on the dive, and I'll make a video about it, just a 1 minute to 1.5 minute long video. So yeah, it's a lot of fun. I really enjoy sharing what we find with the public.
Awesome. Thank you, Anna.
Thank you, Deb.
And then to finish it off, I'm Deb Glickson. I'm with the National Academies of Sciences, Engineering, and Medicine, and I'm the geology lead for 2206. And Thomas?
And I'm Thomas Morrow, Expedition Coordinator for NOAA Ocean Exploration. If you've been tuning in along with us, we've also had Joanna Xavier, our biology lead. She is currently engaged in live interaction, so we periodically have short calls or interactions with students or other interested groups to have kind of a focused conversation about what we're doing and why, and get direct interaction with those communities. But now I have to look at this fish. Yes.
It's not even just the fish. What's in front of it? Is it?
Looks like a plastic bottle.
Bottle? Yeah. Okay, so now we can look at the fish and the bottle.
Ooh.
And a shrimp or a lobster in the top right. And look, that bottle has just become a place for many things to live. Wow. Sorry, I didn't mean to cut you off, Thomas.
No, it was good. No, it's
excited by the fish.
A very bright fish. Mm-hmm. Putting on quite the show for us.
And I'm just going to be completely frank right now. We have 2 geologists here in the back, Thomas and me. And so fish is going to be our identification until someone tells us that it's something else.
The chat room has also asked if this is normal behavior for this fish.
We have seen a lot of fish that do move vertically, but sometimes they're also being disturbed by the lights of the ROV. So they could be disoriented. That might be why they're moving vertically, but it's also, I believe it's a feature that they sometimes use, not necessarily this type of fish, but they sometimes use for hunting. So we saw it on one of our last couple of dives with the cutlass fish, where they were routinely standing vertically in the water column. And as we continue upslope, we're still seeing just a lot of sediment, which is really not unexpected, with some patchy little outcrops of rock. Look at this plastic bottle. It's so interesting. There's so much inside of it.
Yeah, maybe it's a glass
creates,
bottle. Yeah.
Yeah, glass or plastic, and the things are definitely moving. They look like hermit crabs.
There's
There's
a
bunch of hermit crabs
in here.
Yeah, and a worm.
Why are they all inside this bottle?
Because they're safe there.
I mean, sure.
But look at that is a lot of hermit crabs.
Seriously.
Wow.
Maybe they're all just hoping to grow large enough to take the bottle on as a home.
I think the problem would be, though, if they grow too big and they can't get out of get out of the bottle. It looks like there's quite a number of shells that they could be collecting and moving through, though.
Oh, there's one or two of them outside, too.
Yeah, this is really cool. I think it is glass. I'm not sure. I don't think it's a plastic bottle. I think it's a glass bottle.
I think you're right.
And, yeah, there's a hermit crab on the outside.
A couple of them.
Yeah. I don't think I've ever seen this many hermit crabs in one place before.
There's another hermit crab in the...
Two here on the bottom. There was one up to the top left.
Top left of the bottle, who is kind of giving us a nice view. You know what? I wonder if... Okay, here's my idea. This is a jar full of shells.
I think so, too, actually.
That somebody has cast away, returned to the ocean.
Although you could also argue, so I agree with you, but you could also argue that these hermit crabs have brought shells in, and then they have switched them out as they've gotten bigger. So we don't know if it was already filled with shells or if they have found it a convenient habitat to switch out their shells, their body plans. Yeah. Instead of a ship in a bottle, we have hermit crabs in a bottle. Do you think the ones on the outside are like, why am I not inside?
Yeah.
This is very, very cool, though. And it really does show that when you give a habitat to this area, even if it's not a traditional habitat, it's going to get utilized. Whoa.
Not to encourage putting more glass bottles in the ocean for hermit crabs to live in.
No, absolutely not. I'm just saying that if they're already there.
Right.
Yeah. But yeah, there were two more hermit crabs on this side. So what, we're up to about at least 10 that we've seen. That is a lot of hermit crabs. Very cool. Very, very cool.
So pilots are trying to identify, there's a squid floating.
Squid?
It appears.
I need to be told where the squid is.
We can see it on the Sirius view, and so the
There
pilots
it
are
is.
trying to get a good look at it.
Wow. It's kind of far away from us, though. Oh, look at that.
Wow.
How cool. So is this typical behavior for a squid? It looks like it's walking along the bottom.
Well, I imagine there's good things to eat down in the sediment.
Look at that. What a beautiful view. Amazing. Yeah, I really wonder what it's doing when it's sort of bouncing off the seafloor, whether it's, using that as a sensor or... It would be awesome if a fish swam by right now. So one of our shoreside scientists, Kim Galvez, is suggesting that perhaps this is a defense posture. Just amazing.
Fantastic views.
Mm-hmm.
Wow.
It's always great when it's against the blue water, dark water in the back.
Mm-hmm.
It's even more. Yep. Wow. Really quite beautiful. And it's unfortunate, one of our onshore scientists who normally joins us for the chats is an expert in squids and octopus, but unfortunately he is not with us today. Now it's heading back down to the seafloor. Ooh. It seemed very confused to have gotten to the seafloor.
And now he's going to
All
take
right.
off.
Well, that was an amazing view of that squid.
So long, squid.
You can see how zoomed in we were. We were really, really far out.
Yeah.
What beautiful imagery.
So as we're conducting our current move, I'm going to spotlight a little bit on our current ROV pilot. Today was a very exciting day until recently. The Artemis rocket that was going to be launched by NASA, the launch was canceled. And I feel like if I was at home, I would be switching between the launch stream and the Okeanos stream personally. But our ROV pilot right now, John, has worked on the Artemis project. And so it's really exciting to always have that talent come and join us at sea.
From the deep sea to outer space.
Yeah.
It's quite a wide range of areas to work in.
Yeah. Ooh, we have more vertical
So are
fish.
all these fish vertical? This must be a typical behavior for them.
When in Lang Bank. Wow.
Wow. This is really cool. Gonna wait to see if maybe Michelle Sharer, who's in our chat room and is a fish expert, might know what kinds of fish these are. Oh. This is really cool. It's like a fish chandelier. Yep. What an amazing view. A beautiful silvery fish. And head down the whole time. And one of our shoreside scientists, Asako Matsumoto, just said that this reminds her of the Harry Potter party scene where you have all the lights just floating in the air. I think she might be onto something.
It's really interesting how the fish keeps rotating.
Yeah,
it's kind
you're
of
right.
Hunting.
Mm-hmm.
This is a food searching mechanism.
Very interesting. Thanks, pilots, for following that fish. Here's another of that same type of fish. As Thomas was mentioning, it does keep turning in a slightly regular pattern.
Now, earlier in the dive, we saw some fish that were darting towards the seafloor, and we speculated that it was because the ROV was startling them, and that's a defense mechanism. But now I wonder, with this, it looks like a hunting kind of activity. If that's what they're waiting on, if they spot something tasty, they'll shoot down to the seafloor and see if they can grab it.
It doesn't mean that both can't be right, though. These fish might be in a predatory,
yes,
hunting phase, and the other ones may have been disoriented by the ROV, which we have seen before. But I think it's probably dependent on whether it's the same species.
Mm-hmm.
This one's really going down to eat. It's like a little pogo stick.
Mm-hmm.
Bloop. I feel like this one might need some sound effects.
I know. Our video team does a really great job of getting some inspiring audio to back these videos. But every once in a while, I want whimsy. I want something just funny, because some creatures behave in a funny way.
Yep. Bloop.
Yep.
These fish are just simply fascinating. I feel like we probably passed over some very interesting stuff on the seafloor that we haven't talked about because we've been looking at fish, which is totally okay. And we're still moving up slope, and we're moving roughly due south because we were on the northern part of Lang Bank, moving up the slope. and we're at 618 meters now. So getting closer to that zone where we think there might be more fish than we have seen thus far. What do we think? I think that's a holothurian in the top left corner.
Yeah, looks like it.
What just landed on the seafloor? Is that a fish?
Oh, yeah. Zoomed away.
Probably another large dead sponge, which provides a great habitat for other animals. And the sediment is really highly bioturbated. You can see that the different colors. Just a very quick one. So definitely a sponge. I think actually even a sponge that we have seen before on this dive has very similar features. I'm kind of surprised they're not colonized more. I would have expected this would be a wonderful home for some of our deep sea creatures.
I bet if we could open it up, maybe we'd find some more stuff tunneling inside.
Those look like serpulid worms again. So the fish that we had just been seeing, where we saw probably 5 or 6 of them that were vertical, may have been Mueller's pearl-side species of hatchetfish. We have seen hatchetfish before, so this is the Mueller's pearl-side hatchetfish. That is a very big sponge. And so one of the, I think, intriguing questions of our dive thus far has been that we've seen almost entirely large dead sponge instead of small alive sponges, and where these old sponges could have come from. And so I think the prevailing idea would be that right now it's probably coming from somewhere further upslope and that they've rolled down the slope. But we'll find out, hopefully, if we can make it to the top of the slope. And we are slowly moving our way upslope, stopping to enjoy all the things that we've getting to see. We were having a lively discussion earlier about every time we see something dark on the seafloor, do we just assume it's a holothurian? Maybe it's a sea urchin. Maybe it's a bottle. We don't know. Can we get a snap zoom on the one on the left center right now? Thank you. Is it a bottle? It's a bottle. Chances are 1 in 3. Looks like a scallop on the bottle, and it looks actually that scallop is alive. You can see the tissue between its 2 shells.
Cool.
It is really cool. Oh, it just closed up. Did you see that? That was awesome. So it was out, I assume, filter feeding, and then it just sucked itself back in. It looks like there are shells in that bottle, too, so I wonder if there's a hermit crab in there as well. And while, of course, we would probably much rather you recycle your bottles on land, at least these are being repurposed into something valuable. Habitat for our hermit crab friends. That was really cool. So evidently that behavior where they immediately shut their shell is actually called clamming up. I'm not sure if I believe that. I think maybe it's a joke. All right, and as we continue upslope, still quite sedimented, still quite bioturbated, and that bioturbation is when animals, benthic animals on the seafloor, they change the structure of the seafloor itself, whether that's through burrowing down into the sediment and then they leave little mounds or piles behind, whether it's through traces where they're walking across the sediment and they leave tracks, which we saw earlier. So this is certainly disturbed seafloor.
There's a sea star.
See a sea star and more of the worms.
And some shrimp.
I think these might actually be rocks and not a sponge. I don't think I've ever had to try to decide whether things were rocks or sponges before. This may be a new thing for me. And 2 shrimp guarding their area here. So we have a sea star. We have crinoids. We have, is that a fish in the back?
I think a fish and a crab.
And I feel like this is a sea star that we have seen before, but I am not going to try to remember what its name was. And either an anemone or a crinoid. And so the joke that I just made about clamming up, evidently that is a real, actual behavioral word. I'll believe it. Is this one? Oh, this is Ophidiaster. And we have seen one of these before. And in fact, I think the one that we saw was a 4-armed Ophidiaster. And this one has at least 4.5 arms. I'm not really quite sure what's happening to the back arm there. And these are sitting on a piece of limestone. And I will be the first person to tell you I don't know what this very beautiful white thing is. I don't know if it's a crinoid. I don't know if it's an anemone. I'm waiting for someone to tell me. But it is beautiful. And it really just goes to show, again, that you have these small pieces of hard bottom. You get these small rocks. You just find one place, and then they are just covered in sediment. No, covered in biota, not sediment, although the sediment's all around them. Is that a crab? Is it another one of the carrier crabs? It is.
Oh, yeah.
I wonder if this would be a good place to get a water sample.
I think, yeah, John would probably appreciate that.
Yeah.
It looks like it's
Wonder
got
if this-
another sponge on its back.
It is carrying what looks like a sponge on its back. And being the expert sponge biologist that I am, I'm going to go with it's a dead sponge, but I'm not really sure if that's true or not. Our sponge biologist, Joanna, who's our biology science lead on the cruise, is actually right now having a conversation with school children in the Azores. And it's quite fascinating, but she would be able to tell us what kind of sponge that was. Maybe we'll hold off on the water sample as we go up, see if we see something more exciting. Oh, look at that. So that's a couple of shrimp that we talked about that were guarding the area, it looked like. And then this sea urchin that has these very long, very flexible spines and a little test, and it's moving, which I guess is not surprising. And then is that a small coral up to the left?
Yeah. Oh, and there's a squat lobster living inside it.
Boy, every time we look, there's just something more interesting here. And evidently there's lots of little bryozoan colonies that are on these rocks. And the thing that is to the left of this urchin may be a Chrysogorgia. And Chris Ma, our echinoderm expert, is probably going to call in and talk about that urchin while we look at this coral. So this might be a Chrysogorgia. I don't think we've seen one on this dive yet.
No,
I think this is the first.
Yeah.
And it does, it does have a squat lobster in it.
Yeah. Look at those eyes.
Hi,
Hey,
Chris.
this is Chris Ma.
Chris, can you
Hello?
tell us a little bit about the sea urchin that we were just looking at?
Yeah, so this is actually one of my kind of go-to favorites. This is what's called an Aspito Diatomated urchin, and these are really cool because the spines that come off of the animal's surface, they almost act as kind of the sea urchin analog as kind of a daddy longlegs or a spider. It moves using them in a kind of again, in a very almost arachnid way. I think this one is maybe Aspidodiadema, but yeah, it turns out that the spines have little like pads on the tips of them, and so it kind of trundles, picks itself up along the seafloor with these long spines and just kind of propels itself. Just kind of moving along the bottom. So in that sense, it's really weird. These are related, or at least they were at one time thought to be related to diatomated urchins, the long spiny things that we see on in the shallow waters. Once in a blue moon, we get to see an Aspito diatomated with what's called an ale bulb. There's kind of a round bulb that's right on the surface where it'll emit all of its poop. And so, that's like a 2-for-1 when we get that whole package. But they're just so odd looking and so interesting that they're always worth calling in when I see one. That sea star that we just saw over to the right is Ophidiaster, and that you were correct in your earlier narrative that is in fact the same kind of sea star we saw with the 4 arms the other day. This is typically a species that occurs in more shallower water, in reef-type habitats and so forth. And so it's one of these kind of odd reef-type species that gets really deep in this area. But there are several deep-sea species of this genus all around. So anyway, that's a really nice moment. Thanks very much for stopping and getting some nice Aspito diatomated imagery. Those are always appreciated, and one of these days someone's going to come along and write the big book of Aspito diatomatids, and I'm sure they'll be grateful.
Thank you so much, Chris. So Aspito diatomated, I think.
Always a pleasure. Thank you.
Yep.
Deb. Bye-bye. What was that?
I'm trying to pronounce Aspito diatomated. Bye. And so we're seeing
all right. Bye-bye.
We're seeing a few more little rocks. I mean, I don't even think you can call these outcrops. They're just little rocks. And is it a lobster? Is it a crab? Might be a crab. I don't know. We'll find out. But, our goal is to continually move up the slope and take some really nice imagery as we do. This dive is a little different than some of our other dives in that we will not be sampling on this dive because we're within 12 nautical miles.
Ooh.
So is that a
those spines?
Is it a crab? It's very spiny. That's about as far as I can go in my identification. How cool. Oh, and look at what it's sitting on. You can see, I think, some of the fossil coral in this rock. I know we're supposed to be looking at the crab.
Tell us about the rock.
A lithode crab. Lithode crab. Probably because it looks stony on the outside. Beautiful. But I do feel that the rock that you're sitting on, that this crab is sitting on right now, looks like it has a lot of fossil coral in it. This guy is like chowing down.
Yeah.
Look at the top right of this. I actually don't know. I don't know what this is. I'm not carbonate geologist, so I'm not sure what this is, but it certainly looks like a colony of corals, of fossil corals. So I'm hoping that somebody might want to tell me what this is, but what a beautiful rock that we've stopped on. It does look like a fossilized reef. I don't know what species those would be that would have made up that reef. But just a beautiful image. What do we think that grayish blobby thing is on top of the crab, above the crab? Maybe it is a bivalve. Pilots, could we get the lasers when you have a chance? Yeah, I'm not sure if it's a bivalve, if it's alive. I have no idea. But what a beautiful image. So the crab itself is about 10 centimeters from leg to leg, as it were. And that also gives us an idea of how big those little fossil corals are as well. That's really helpful. I get the feeling that crab is not super thrilled with us continuing to watch it. Oh, so this could also be Monstera cavernosa, which is a type of coral. So it's a fossil scleractinian coral, and that's just really cool. That's the first one that we've seen, I think, that's been so well defined, that wasn't covered in sediment. It's just a fantastic example of what the underlying rocks may be in this area, which is mostly fossilized reef. So very, very cool. So a lithode crab, which I don't think I've ever seen before. It's very stony looking body. It's bumpy, spiny looking body. And we're at about 609 meters, moving upslope, slowly but consistently. That's what we're doing today. And what looks like maybe crinoids. And there was probably a sea urchin test off to the right. Oh yeah, definitely looks like a crinoid. And another shrimp. And a stocky thing that I don't know what it is.
All right. For those of you just joining us, we are doing our usual lunch change-outs where we try and get folks a plate of food. So currently we have myself, Thomas Morrow, Expedition Coordinator, and Sam Candio, our mapping lead, on board while the science leads go and grab some food. I think Sam has a stronger background in biological things than I do.
Only slightly.
But I can identify this as oh, no, I take that back, because I've looked at things and said they're anemones before, and then I've been corrected and told they're corals. Let's check in with the chat and see what the chat says this is.
Have we lost our science discussion?
All right, so no discussion in the chat yet on what kind of anemone this is. There's a nice red color in the middle there. And it looks like it's sitting on a small outcropping piece of, it's hard to tell if that's rock or sponge. We've seen a lot of rock-looking sponges that have fallen down from higher up.
I think that looks like rock to me. Yeah, for sure.
So in this hour's edition of Sponge or Rock, we have rock coming up as the answer.
Oh, look at the pattern on the base of it.
So you may have noticed some lighting changes while we were getting a few good shots of this anemone. And if you switch over to the Sirius cam, you'll see there are a couple of arms sticking off of D2, and the videographers and the engineers work in concert to get these lights oriented in ways that give us really good shots that are both beautiful to look at, but also sometimes really critical for identifying organisms. Especially with some of these underwater creatures, there are really small details that scientists have to pick out to positively identify something.
Looks like we might have another glass bottle. I saw as I was coming in the glass that was full of hermit crabs.
Yes.
Pretty wild.
Yes, and actually one of our onshore scientists commented in the chat that a lot of those shells looked different than what we had seen locally, so it's possible that was literally someone's shell collection that had gone overboard at some point.
Interesting.
And then was colonized.
Yeah. Life always finds a way. Now, is this half of an urchin test?
I think so.
Looks
We
like it
were
might
looking
be.
at a dead one earlier and speculating on some tracks in the sand.
I think that one's been gone for a little bit of time.
We are still hoping to find a live one. We've been asked to look for what looked like just a little bun on the seafloor, a little piece of bread.
Looking for bread. This looks like the same anemone we were looking at before.
I don't think we need another zoom on this. Thanks.
Little hole. Anything living in there? A little shrimp.
Yeah, I'm surprised we're not seeing more. Generally, anytime we found something that was shelter on this dive, it's been actively colonized.
Yeah, it looks like a nice little house. We got a couple of anemones guarding the entryway.
So if you're just joining us, we are diving on Lang Bank, which is a feature on the east side of St. Croix. We started right around 700 meters depth, and we're heading up towards roughly 400 meters. And the target of this dive is really focused on the different fish aggregations we may come across. This is an area where there are a lot of fish that are commercially important species. And one of the big questions from the scientific community was, how deep exactly do these fish go? Are we likely to see them at depth? So we've gone a little deeper than they're known to be, and then we're kind of approaching their typical habitat range from below.
And that's part of the intrigue of this dive. There have been an abundance of observations on Lang Bank itself in the shallows. I think there's actually another vessel working there right now. But having that continuity between what we're seeing in the shallows and then what we're seeing at depth, I think is a very interesting concept. And whether it be similar species that we're seeing, like having depth extensions, or just seeing how different communities thrive at different depths and seeing where that shift is, I think is extremely interesting. Oh, is this one of those urchins? Is this the bread we're looking for?
I think so.
It does not look alive.
No.
Looks like we have a fish coming center screen. Oh, there he goes. Little camera shy. Looks like we have another jar. Looks like a jar. Looks like there's some sort of spine up on the rock in the upper left as well. It's kind of hard to see if anything is making this jar its home. Looks like we have another bottle and a couple more anemones on this small little rock outcrop.
Maybe a sponge back there.
Maybe.
Ooh. What is this? What is this creature off to the right?
Looks like we have a shrimp.
Yeah. And then there's a fish to the left. Again, just the tiniest outcrop, the tiniest piece of shelter or hard space to anchor, and suddenly we have this big diversity of life.
Okay, and I'm back after a really lively live interaction with students back in the Azores, San Miguel Island, joining us from Expo Lab. Oh, and it seems that we have quite a lot more biodiversity here now.
We just got to it.
Okay.
Oh, did you see that anemone in the back?
Oh, there in the back?
It just retracted its tentacles a second ago.
You mean the white one there to the left,
Yes,
top left? Okay.
yes. Yeah, when we first came up to it, it was out, and then I just saw it react to
and
something.
we've seen also these other anemones that are here at the front. We've seen them also in the previous dives with these very long, tall bodies. Oh, look at that! Yeah, beautiful tentacles there. And there's something else pinkish that I can't quite make what it is.
Yeah, the center.
Yeah.
Yeah.
Oh, and these strange carbonate blocks, isn't it? Very round shaped. I wonder if those are fragments of corals that have.
My guess was some
gotten
kind
here
of sponge, but
Oh, you thought?
it was a guess. It was totally
No,
a
I
guess.
didn't.
You are the expert.
No, I don't think it is. It doesn't look like a sponge to me. Oh, and some bottles as well. So we are still on a relatively flat area. Now at 585 meters depth. And we hope to get as shallow as 300 meters throughout this dive. But we've seen already a variety of both fish, some corals, sea urchins.
Yep, we'll have to check this urchin
Oh,
again.
yeah.
Oh,
Is that just a test? Yeah. Ooh, a little tiny fish that apparently was living underneath it. Oh, look at that.
quick little look at this.
It is really small.
Yeah. I say I wouldn't even put the lasers on. You might cover it up. It's so tiny.
But a very pretty little guy. Look at that.
Yeah, a centimeter, maybe two. So Chris Ma is identifying this as a different urchin than the Conolampus that we were looking at, or the evidence of Conolampus that we were looking at earlier.
Oh, is that yeah, it's a ctenophore that we're seeing coming into view now. It's rather difficult to see because it's so...
Yeah, against the sediment.
Yeah. Yeah, and George Matsumoto here in the chat room was saying that that ctenophore was in the genus Bathocero. So I think we've seen also that earlier on in this expedition. Oh, that's a brittle star there moving right at the bottom. Yeah. She's like, feels like she's stretching.
It's very bright pink color.
It is.
Bubblegum pink.
Yeah. I'm not sure if you guys saw this one previously while I was away. But
I don't think so.
No, this is very,
At
very
least
big.
not from a coloration standpoint.
Okay.
I don't remember us seeing the same color.
Yeah. Would be great to have a closer look at it, the central disc, as that might be very informative here for the folks in the chat room.
Chris thinks it might be a Bathopectenora heros.
Oh. So it's roughly at 12, 15 centimeters from arm tip to arm tip. Yeah, and Chris here is commenting here in the chat room that seems that all the ophiuroids here are rather big.
Does that have something to do with the depth range that we're at?
Yeah, I'm not sure.
This time I feel like that one's a bottle and not a holothurian.
Yeah, no, that's definitely a bottle.
That's my new. That's going to be my new game for the day. Is it a holothurian or is it a bottle?
Earlier, Sam and I were playing Sponge or Rock.
Yeah, Sponge or Rock is another one that we've been playing on this particular dive. I think that we were still interested, Joanna, about the fact that all we've seen mostly are these very big
large
dead sponges. And so we're wondering if, are we going to move up slope and find where those sponges are alive?
Yeah, I really am hoping so. And especially those, the sponges that we've been seeing so far, I think they're all typical of much shallower depths.
Mm-hmm.
So I wonder if actually they could be all the way out here because they get ripped off from the reef because of, bad weather or hurricanes. And then maybe they get washed further ashore and then they just sink. Because I think some of the skeletons of those sponges that we've been seeing so far, they are very typical of much shallower water.
What do you mean when you say much shallower? What depth range are you talking about?
I would say down to maximum 100 meters depth.
Oh, wow.
But most typically down to 50 meters depth. So
much shallower than where we are right now.
Much shallower.
Interesting.
And I mean, and we know the area is, it's not so uncommon to have these hurricanes going by here in the Caribbean region. And you can imagine that there's the impact of that on the marine system, especially the shallow water ones, must be quite substantial. So it wouldn't surprise me if some of these do get ripped off from the reefs when such extreme events occur, and then they just get washed ashore, sinking to the seafloor in areas where they don't thrive nearly as well.
That makes a lot of sense. So we're at 575 meters now, so we are getting closer to the depth range that we expect to see some of these commercially important fisheries species in.
Yeah, we have again an urchin here, center of the screen. I think you've come across it earlier on, this species, but I was not.
Yeah, if we zoom in on this one, if it has little spines that look like Q-tips and other little spines that look like tubes, it was something we've seen before, or at least I think it might have been. But
let's have here a zoom in on it. Very bright red test with those white lines there.
I'm not sure this is the same one that we saw before. This doesn't seem to have those specialized spines that the other one did, although I will leave it to our echinoderm expert to prove me wrong. But this does not look like the same one that we had seen previously. To me. And only to me. It's another beautiful sea urchin. Yes, we are getting confirmation that this is not the same sea urchin that we saw earlier. We can see a lot of the traces of animals walking on the seafloor, moving along the seafloor. What is that?
Also trying to figure out. I'm wondering if this could be like also an urchin. Yeah,
that would be my guess too.
It does look like the type like a sand dollar urchin.
Like a sand dollar, yeah. But that's what I was wondering as well.
At least the surface seemed to have that sort of structure. Oh, it's small. It's like, yeah.
And so, as we've mentioned, we're moving upslope to the south. We're on the northern part of Lang Bank. And this is a very large carbonate platform. It has sort of the lower levels of this bank are made up of elkhorn coral and other parts of the corals, and then it turns into sand and sediment rubble. So it's a big pile of limestone and sand. A lot of it's biogenic, and then a lot of it has open spaces because it's a coral. There are lots of void spaces in it as well. And we're just looking at the surface expression of some sediment cover, all of which is pretty bioturbated. So there's definitely things living on it or within it, or both. Oh, that last thing that we looked at that we thought might be some kind of sand dollar type urchin stumped our echinoderm expert.
Oh. So if Chris does not know what it is,
then we definitely didn't know what it was.
No.
And Lang Bank is it's on the eastern side of St. Croix, which is where we are right now, and it's at least 15 kilometers wide. It's a really
large
big,
bank.
flat bank with an average depth of around 20 to 30 meters.
Yeah,
so
and part of it is actually a protected area because it is known to be an important area for... we just saw... What was that?
I don't know. I don't know what that was.
It's a very important area as a spawning where you can find spawning aggregations of a number of commercial fish species.
What's that yellow thing, Joanna?
Let's have a look at it. Well, just from the general appearance, I would say a sea cucumber, but that's actually a very unusual. Ooh.
Well, that's weird looking.
What is that?
I don't know.
It looks like to have these tentacles like pompon anemone, but I've never seen it in this color. Oh, that is unusual. I wonder if this could be still an anemone with this. Nolothurian with this.
I don't see why it couldn't be. It's too bad it doesn't want to roll over for us so that we can see more of its body plan.
Yeah, exactly, because I also don't see, the typically in the anemones, well, the pompon anemones, the mouth is very difficult to see because there's so many of these tentacles, which is why it's called a pompon anemone.
Right.
So I think that's it's not moving, so might very well be one of these pompon anemones.
If only we could just poke it a little bit to see.
Yeah, we cannot.
We cannot poke. But actually, looking at the sediment around it, you can also see it's just rubble, right? It's all biogenic rubble. You see coral rubble. There's like casts, there's pteropods,
shells.
So
fragments of corals and
it is just a pile of biogenic sediment. How interesting.
Yeah. And intriguing, this one. Very intriguing. Oh,
what keeps jumping? What is that? What does it jump mean? What was that?
My guess it's one type of isopod that lives buried on the sediment.
Oh, then we don't need to keep following it. That's okay then. No, just kidding. So look at this. This shrimp has a nice burrow.
Yeah.
It's found some void spaces in the underlying rock. I guess it's 3 shrimp.
And it's interesting because you can see there, like at the margin of that burrow, you can see how you have all of these like serpulid worms, tubes, and bits of probably coral, all very cemented.
Yes.
Together.
What is that white thing that we're zooming in on right now? Oh. What is this? It looks like some sea urchin shell, spines.
Agglutinated for some reason.
Is there anything live in there?
Doesn't look like.
Oh, and a little hermit crab. It's awesome because when you zoom in on these features, which don't look like they have a lot of life from this far away, when you zoom in on them, you really see so much more.
Yeah, and while we are not able to collect any of these organisms to confirm what they might be, and here we have a really nice anemone.
So this one we saw earlier, and we did not know what it was.
It's an anemone.
Okay. That one we were not sure about.
It might actually be a Syriantharian because I seem to see that there are 2 rows of tentacles. So that would be a tube anemone. So part of it actually lives buried into the sediment. Oh, that's a very pretty one.
What is that off to its left that we're focusing on?
It's a bristle worm. And one of those serpulids we've seen earlier. Some of them are very pretty because they have these tentacles that are striped.
I was going to say, most of the ones we've seen so far look like they were like a white or very pale, almost translucent.
Mm-hmm. Oh, and we have here a sea star. Also seems to be a relatively large one.
Yep, another sea urchin, another brittle star.
Yeah. Sea star, this one now, a large one.
Oh, you're right, a sea star. So sorry. And look at the bioturbation that's up to the right of
there's makes a little...
yeah, it's circular.
Yeah.
Some of these trace fossils that we find on the seafloor, the traces of the animals that are walking around, are just beautiful.
Oh, look at that.
You can see its little
and
tube
you can
feet.
see tube feet. And the ornamentation of these, of the arms, and it's just amazing.
What is the white thing that's on the left of this sea star? Is it just sediment?
Hey, this is Chris Ma.
Hi, Chris.
Hello. So you have here one of the, surprisingly enough, in collections, this is one of the most abundantly occurring species, but whenever we do the tropical Atlantic Okeanos dives, I think we maybe see one per, like, the entire mission.
Oh, really?
This is Nymphaster. So this is called Nymphaster Erinatus. This particular form is called it's called Nymphaster subspinosus, but is arguably a subspecies of Erinatus or a synonym of Erinatus. So it's hard to say. But one of the nice things that identifies this species is the fact that the arms. You see how the plates around the edge kind of join and form these long, almost arrow-like points. That's the characteristic of this species, and it's really interesting because it's thought that, well, at least people have argued, and well, when I say the starfish community, I mean me and maybe one or two other guys, that they have produced this morphology to act as kind of a snowshoe, like it creates more surface area so that they can operate on these soft sediment surfaces. This particular genus, Nymphaster, is actually seen in the fossil record from the Mesozoic.
Oh, wow.
So seeing these alive, I mean, there's some debate whether it's the same thing because a lot of the European fossils are kind of are based on ossicles, individual pieces, rather than the whole thing. So the individual pieces look very much like this, but and some of the few body fossils do kind of look like it, but again, it's an active area of research. But yeah, I mean, it's a beautiful animal when we're seeing it alive. I've seen the specimens in the museum. From what I've observed of the few times we've seen them alive, they feed on detritus. They aren't necessarily predatory, but they do go after, for example, dead squid. Mm-hmm. I've seen them bury themselves into sediment, and there are even some scale worms that live in association with them. So thank you very much. There's probably some paleontologist who's overjoyed that we're seeing one of these alive.
That's excellent. Thank you, Chris.
I unfortunately... Yeah. Oh, is that an urchin on the left? That little trail? Well, that's fine. That's okay. We passed it.
You have
anyway,
a much more trained eye than we do, Chris.
Well, that's both a blessing and a curse, believe me. Anyway, I'll let you guys go. Thanks so much for stopping.
Thank you. That was great. So it seems that it was actually a rare finding we got there with that Nymphaster.
Although he said it was
common,
it's actually supposed to be common.
Yeah,
just haven't seen very many of them.
Yeah, in during these ROV dives, yeah.
Mm-hmm. But yeah, as Chris was mentioning and as others have mentioned in the chat room, the sediment here is highly disturbed. You can see lots of tracks of animals. You can see lots of things that might have been burrows or mounds. There's really a lot going on the seafloor. And then there's so much of that biogenic sediment that when we zoom in on animals, we tend to see these broken up corals. We see pteropods. It's really very interesting.
Yeah, it would be great to see some more of the animals that are actually making these burrows and these mounds. Oh, we see again one of these, what I think is an anemone. This yellow one, this pompon.
Oh, yeah.
Yeah. Let's have, again, just a quick zoom on it. See if we can find any other character that would help us. I remember seeing some of these pompon anemones in other areas, but not yellow like this. Look at that.
It's really cool.
It is.
Ooh.
Just so many tentacles. So you can, yeah, I think my best guess really is that this is a pom-pom anemone. Yeah, I think I'm convinced that this is a pom-pom anemone right by now.
I will believe you. So moving right along, we do see small outcrops as we've been going along. We see these small rocky outcrops, although half of them have been sponges. No, but some of them are actually, they're either fossilized coral, they're old limestone. Ooh, this one has a couple of shrimps, a tube anemone, something else. Is that another urchin back there? Can't tell from this far.
Yeah.
Oh, look, the tube anemone just retracted.
Oh, it did?
Yeah, I think it's because there's
the shrimp touched it.
Is that a shrimp?
Yeah. Yeah, that is a shrimp. It just touched it there.
Yeah, and it did not like that. There's another shrimp.
Yeah. They seem to really like the crevices of these.
There's actually two. There's one right off the screen to the left, I think, as well.
And we have a small scleractinian coral, a solitary coral there, also right in the front. Oh, and a few more actually on top of this carbonate plate.
Yeah, there's actually several of them.
Yeah, there's a few of them.
I think that thing you saw in the background was another pom-pom.
Yeah.
There's another one right there. So as we keep moving up slope, I'm just very interested by the, it's like we are seeing actually a high diversity of life. It's not hugely abundant until we zoom in, and then all of a sudden you see more.
Yeah, it's not very dense, but... And of course, not surprisingly, because we haven't been seeing so much hard substrate here, we haven't been finding a lot of corals.
That's another
look at that.
Oh, and is that a coral to the right?
There is one of those small scleractinian corals there to the right.
What's to the left that's very bright white against the sediment? Looks like there's a chain of them going up to the left.
Could that be just a fragments of urchin? Yeah, I think it's a broken urchin test.
Really?
This one here, at least.
Oh.
Really looks like it. Yeah, I think this is and I think this is an empty urchin test as well, to the left.
You can see as we move up, we keep seeing this bioturbated sediment. We see the darker and the lighter, and whenever we zoom in on the darker area, it's just filled with rubble of animals. We see pteropod shells.
Bits of coral, some bits of bivalves, fragments of urchin tests, or some spines. Yeah. Different types of urchins here, actually, just on the view here. Maybe worth having a quick zoom on that larger one, the white one there, center. Not sure if it's alive, but
it looks like it's one of those urchins.
It stands out for being that big.
I think it is a live version of the urchins that we were looking at earlier.
Yeah.
That one that Chris Moss said looked like a scrubbing bubble.
And that formed these trails.
Yep. It does look different than the one we saw earlier.
Yeah.
He's calling it Conolampus, which is not I don't think that's the same one we did see earlier.
Conolampus.
Conolampus.
Yeah.
Maybe it is then, yeah. It does.
But it doesn't.
Yeah, it's alive. Yep.
Oh, it is alive.
Yeah, so this is earlier in the dive, for those of you who weren't here with us earlier in the dive, we actually saw two of these, and one was sitting right in the middle of a huge track of trace fossils that it had made. And we could see it on camera too when we were looking down from Sirius onto the Deep Discoverer. But then that one and the one we saw after were both dead, and this is a live version of that same thing. And Chris was saying that the tube feet are, I think they're all on the bottom, and that the spines were very reduced, and indeed they look. We can barely see any spines on the surface of it. But looking here from the Sirius view, we don't see nearly the same sort of trace network that we saw previously. And here's another one of these urchins. I think we've seen a greater diversity of urchins on this dive than we have anything else yet.
Yeah, it is a very diverse area, at least for echinoderms.
This looks like the one we just saw a few minutes ago.
For which we don't have yet an identification.
Exactly.
Seems to be something unusual. And so we're now exactly at 550 meters depth. We've started at roughly around 700 meters and hope to continue to ascend up to 300 meters depth on this dive.
And the temperature is about 11 degrees Celsius here. And this is dive 7 of the 2206 Voyage to the Ridge. This is our third dive in Puerto Rico and the U.S. Virgin Islands, the EEZ around that area.
And we are
and
here.
Look at the traces. You can see all the traces off to the right.
Some of them, yeah. Yeah, and we are here in a very interesting area on Lang Bank, which is just the northeastern tip offshore of St. Croix. And this is a very important area because it's an area known to serve as a spawning area for a number of commercial fish species, such as the red end and a number of groupers. That's a sea star
of some type,
with puffy and with plenty of arms.
Yes, it has many, many arms.
8 arms. And this I don't think we've seen before.
We have not seen it on our dives. It doesn't mean that other expeditions to Puerto Rico and Virgin Islands have not seen it.
Of course.
I think I count 9 arms.
1, 2,
this
3,
reminds
4,
me of
5,
Cocinas
6, 7, 8,
Therias.
9 arms.
Interesting.
Wow. And it's moving towards us.
Oh, look at that.
Does it look angry?
No.
Just kidding. It is beautiful.
It's interesting. The surface of it reminds me of shallower water species that we have up in the Northeast Atlantic called Cocinas Therias.
And it's funny that you say that because to me it looks like ones that you can see in the Pacific Northwest as well.
Oh.
So, which I'm sure is not the same.
Hey.
Hi, Chris.
This is Chris Ma. As if you needed to guess who it was. I'm sorry, I'm not interrupting, am I?
Nope. No. Go ahead.
Okay, so this is Coronaster Briareus, and you're correct. Oh man, that is so beautiful. This is just a love. I've been waiting for so long to see one of these on the Atlantic side, and usually we don't get that deep to see them. Anyway, so Coronaster is a genus of sea star that occurs in the tropical Pacific and Atlantic. We saw them on the Capstone expedition where they looked like giant jumbles of arms, and they're multiple armed. They look different in many different respects, but they all have the same kind of general morphology, which is to say they're these big multi-armed things, and they have all of those. So there are spines along the radial center of the arm, and then there are these side series of spines, and all of these spines have little pom-poms filled with pedicelaria, little claw-like, they look like they're kind of like Brisingids in the sense that their pedicelaria they look like little bear traps, individual pieces that have jaws on them. And so it's not clear what they do with them. I mean, they're either predatory on sort of swimming things that land too close, or they're defensive. In Brisingids, the spine plus pedicelaria assemblage, if you want to call it that, are used for predation. They feed on small swimming like amphipods and things, but these don't have the same kind of behavior as far as I can tell. There are definitely, beyond the generalization that they are probably predatory, I'm not entirely sure what some of these things do, which is why I've been wanting to see them alive, because there's some evidence that they feed on other echinoderms. There's some evidence that they feed on any number of things, but if they do feed on these things, why are they bristling in armament? If that's indeed what these structures are for. So, when we were zoomed in on the surface, and the other thing is that multiple armed sea stars tend to be predatory. So everything about them cries, predator. But we really don't understand anything about them. And this is, how big is this? This is also a nice sized one, I think, isn't it?
We'll just ask the pilots to get to get the lasers
Yeah,
on so that we can
but-
have an idea.
Yeah, but as we're zooming in again, you'll notice. I mean, some of those spines that are bristling, that are on the surface there, those are sharp. Those I've been stung by those. I mean, they're not toxic or anything. They're just sharp. But I've been stung by those through like rubber gloves. And again, when they're menaced, or when they're feeling like they're all of those puffs of pedicularia swell up and rise to the surface, like a battery, like a firing battery. So, they're really neat animals from an architectural standpoint. I've been trying to sort of pin down a study where I could work on these, but there really isn't a lot known about them. And okay, so that was the lasers were on, and it was about, so that's larger than 4 inches, which is like 6,
this is
7,
like
8.
40
so that's a big
centimeters.
animal.
Yeah, it's very large.
Wow, that's a sexy beast. I'm going to have to say that in public. That's amazing. I mean, like I said, we've seen them in the Pacific, but I've never actually seen Briareus before. And for all I know, this is a second species. I really don't know, because the shallower, we've had mesophotic or close to shallower water specimens of Coronaster, and they're generally more delicate, if you want to put it that way. The arms are much less well developed. They're smaller. This is a big one. I mean, this one's really nice. I mean, in the sense that it's very robust. The arms are quite thick. It's very large. I don't know if I've ever seen one that is quite as big as this. So that's amazing. So anyway, it's probably it's its own blessing that we can't collect it. Not that I needed to know what it was further, but it's a fascinating. It was wonderful just to see it.
Yeah.
So and I'm sorry, I don't know what these red urchins are. I mean,
no problem.
I'm pretty sure it's some kind of temnopleurid, but there's a couple of them, and I have to homework this one. But
okay.
I think if we see more of those echinothurids, it'll probably be Ariosoma. So anyway, thank you. I'll talk to you guys later.
Excellent. Thank you, Chris. Yeah, the Sea Star really gets the prize for largest one so far.
Yeah. But also just in general, we've seen a great diversity of the Echinoderms on this particular dive, and their trace fossils that go with them, the burrows and the tracks they make.
Yeah, the biological traces,
Oh, yeah, well, the urchins really have been outstanding today. I don't know I've seen as many of them, and here's yet another one. And but it's also filled with mystery, because almost, even with that one that we just saw, the Conolampus, all of those kind of bun urchins, have all been kind of dead, and I don't know why they're just there, so it really is kind of a mystery. Another one of these red urchins. Okay, I'm going to have to go figure out what this is.
Yeah, because
thanks so much.
All right, thank you.
Thank you, Chris. Yeah, because one would think that...
Joanna, I'm sorry.
No problem. So one would think that these Conolampus urchins...
You were just going to say it's another mystery,
Yeah, that these Conolampus urchins would be, if they would be dug up from the sediment because, they were being predated upon, that you would see them broken. But no, they're pretty intact. They seem pretty
totally intact.
Yeah, they seem pretty intact. So it's it's a mystery.
Exactly. All the evidence would suggest that we should be seeing living ones at some point, and we haven't. I mean, even that one that we just saw, I was looking at the picture afterwards, and there's serpulid tubes on the surface.
Yeah.
So even if it was alive, it's moribund. So anyway, I'll let you get to it. You have a fish, so I'll talk to you later. Thank you.
Thank you, Chris. That was great.
What kind of fish is this one?
Yeah, I do not know. Let's see if we get someone here in the chat room that can help us with the fish identification. Not sure if this could be some sort of this Nazumia fish. Has this dorsal, very long dorsal fin. Oh, it's a grenadier.
Oh.
And another one of those pom-pom anemones there.
Yeah, whatever they are, they're pretty common around here.
They don't seem to be so rare. It was amazing to see actually the tube feet of that large sea star, how they actually coordinate all of those hundreds of tube feet to move around.
It was great that it moved around too.
Yeah,
it was nice to see it
really
dynamic in action.
Yeah.
Here, this is getting a little bit more ripply.
Yeah, exactly. So perhaps this is an area with a bit more current.
I don't know, it does not quite look exactly the same as what we've seen earlier. There's another one of those fish.
Another one of these grenadier fish.
Mm-hmm. So the slope that we're on is mostly, carbonate sediment mixed with, as you've seen, old urchin shells and spines and other biogenic material, broken up coral. But what underlays this is some massive coral reefs. And the area that we're in was built by two main types of coral, the Acropora palmata and the Orbicella faviolata. And the elkhorn coral, the first one, I think is threatened pretty much everywhere it's left. So this is a really important area for fisheries, but it's also very interesting to think about the geologic history of this bank of St. Croix and how this area has formed. Oh, we have an identification for the yellow thing, maybe. Joanna, you want to tell us about?
Yeah, it looks like this has been observed. Oh, so it actually. Okay, so someone just passed on to us here in the chat room actually a video of what we've been calling this pom-pom anemone, and that when they try to touch it, actually it just took off from the seafloor, and it seems to be a terepelid worm. And it looks totally different once it's actually
moving
and up from the seafloor. So I wonder, it might be. It looks very much.
It looks pretty similar.
Pretty similar to what we've just saw.
The video that we're looking at.
It's also in the area, so it was made here in the Anegada Passage, here around Puerto Rico. So wow, so there you go.
Okay, so not a pom-pom anemone.
Not a pom-pom anemone, but rather a terepelid worm in which when it's very still like it was as we've been seeing it.
You know what it looked like? Have you ever seen Star Trek? It looks like a tribble. It looked like a tribble.
Okay.
Like puffy and wiggly, and I guess I'm the only one who watches Star Trek. That's okay. Is this a stalked crinoid?
Yeah, it is.
What's up to the left of it?
I think that's another urchin there. On the move.
Not quite sure I'm seeing the movement, but...
It's very slow, but look, I think now you see.
Oh, there we go. Now I see it moving. So we're zooming in on this crinoid, this stalked crinoid. And one of the things that's endlessly fascinating to me about the crinoids is that they are animals. You know, when I look at them, I think, oh, it's a plant.
Yeah.
But it's not. It's an animal. All of those protrusions along its stalk are really fascinating. Are they to help keep hold it in place for
Probably to
sediment?
hold it in place, yeah. They function a bit like a root. Pilots, can we have a zoom in also on those two sticks that are upright just to the right of the crinoid? We've seen a number of these, yeah. Curious about what they may be. It's difficult.
No idea.
They're quite interesting. It actually reminds me a bit of some of these papillae of the sponges that we've seen previously. But here the sediment seems to be far more consolidated, which means that the body would not be buried into the sediment. So that's it's a mystery there. These tubes. Yeah, you're at the remains of a urchin test.
Oh, look at that. There's another yellow one off to the bottom right, and then
it's a pity. It's a pity it doesn't move from the sediment.
Yeah.
Okay, and this.
Your guess is as good as mine.
No.
Your guess is better
well,
than mine.
I was wrong with the previous pom-pom anemone, so I'm now really wondering.
Do you want to call this one a pom-pom anemone too?
Because that it sure is what it looks like to me.
This does actually look very different than the yellow one now that we're seeing them next to each other.
So this is now probably a liponema, so one of those pom-pom anemones. But I mean, on the other hand, when we just saw the video of the terevelid
you felt pretty confident about that before
too.
Yeah,
I was too.
But this is why exploration is actually so much fun, because you can look at it, you can guess what it might be, but sometimes you're wrong, and that's okay. What are the dark tips on these anemones?
Yeah, I wouldn't know. I don't know exactly why they have this different coloration there on the tip of those tentacles.
So the chat room
some,
is saying Liponema. They agree with you.
Yeah.
Maybe they'll tell us why there are the dark spots on the edges of the tentacles.
But it would be great now if we could actually see one of the terebellid worms in action. Because sitting just like that, it was really difficult to imagine what it could be.
So our chat room scientist, Scott France, who was the biology lead on the previous expedition, 2205, actually does not know what those dark spots would be.
Yeah. Oh, there it is. There is what we thought was another pom-pom anemone, because it looks like it has these very long tentacles.
Yeah. Perfect comparison
Let's
time now.
See. Yeah. So let's see if it actually moves. Yeah. Let's see if it moves. If it takes off from the sediment now.
You'd like that, wouldn't you?
I would really like that, because it really tricked us earlier on.
I will say it does look like the video that you were sent from an earlier Nautilus dive in the Anegada Passage. Morphologically, it looks very, very similar.
Yeah. Yeah, it's still undisturbed. It does not move. It does not want to move. Can we nudge it?
This is why I've always thought there should be a poking stick on the D2. I have been saying this for years, that there should be something that we can just poke the animals with. But I have generally been told that that is not appropriate behavior.
Or just something like a little water jet. You know, just very gentle water jet.
Pilots
we don't really need to
water at them
poke
right
them.
Now.
I feel like if they won't let me poke it with a stick, I'm not sure they'd let you poke it with a water jet either. So have you noticed, Joanna, that in the last, I don't know, at least 50 meters or so, maybe even more, we haven't seen a lot of the dead sponges.
I think it's moving.
Is it moving?
Ooh,
it turned.
Looks like it's
waving
move.
at us.
Oh, wait.
They're putting a little water towards it, I think.
Okay.
Oh, yeah, look at its bottom.
Then you see, then you see the rest of the body. Look at that.
Oh, look at that. I've never heard anybody who's so excited to be wrong.
Yeah. Because now at least we could see it, because it was really hiding underneath.
A polychaete worm, is that how you pronounce it?
Terebellid worm.
Wow, that was awesome.
It sure tricked us, this one. Thank you, that was great.
What a cute little fish.
Oh, beautiful.
Oh,
whoops, that was a very fast fish. Yeah, and I'm really starting to think that these little sticks on the sediment could be some sort of sponge. Or at least the papillae of a sponge that would be living inside the sediment, so the main body would be found inside the sediment.
Oh, you think so?
But I had the feeling that this sediment was a bit more consolidated than what we've seen, for instance, in our previous dive.
I think it might depend on where you are on the sediment.
Yeah, possibly, yeah.
Boy, that is a cute fish.
Yeah, look at that.
Is that the one called the green eye?
So our video engineer, who often knows more about the fish than we do, suggests that this might be the green eye.
The green eye.
See if the... Oh, and yes, that's
green
exactly
eye fish.
what the science team is saying on shore, that they think it's a green eye fish. So thank you to Roland, our video engineer, and to the shore side.
Yeah. Mm-hmm.
I don't really see
Even
the
though
green.
no, me neither. That's what I was thinking.
Looks yellow to me.
Wow, but these colors are just amazing.
Yes, they are. And I mean, this is why it's so valuable to have the onshore science component of what we do, because it allows us to draw in a much greater variety of expertise onto the ship. And so we can have a fisheries expert, we can have an echinoderm expert, we can have other people who can help support the expertise that, of the people on the ship. Yeah. It's really great. Is that another rat tail?
Yeah, without them, these dives would be way more boring.
Yep. Is that swimming backwards?
It looks like, but it's a grenadier, possibly in the genus Nazumia that we've seen also earlier. Yeah, but it is moving.
Oh, that one actually has
backwards.
A different identification in the chat room, unless it was something else.
Oh, okay. Chlorophthalmus agassizi. And it's these beautiful dots on the pectoral fins. It's so pretty.
The chlorophthalmus may have been the green-eyed fish, or is it this one?
Okay.
Do you see what I'm saying? It may be the prior fish. It may not be this fish.
Not this one. Okay.
I don't know. We'll have to see what they say in the chat room.
Look at that.
This one's trying to disturb one of your sticks.
Yeah, I'm more and more convinced this actually belongs to a sponge. Probably will find some sort of sponge body underneath the sediment there. So that's what we would call an endopsamic sponge that lives buried in the sediment.
So we're at 525 meters right now. Mm-hmm. And, this is the area that we were starting to expect to see some of the more commercially important species. We haven't yet seen very many of them.
No.
And so we're just zipping along.
See if we make some distance now.
So maybe we should zoom through this area a little bit until something interesting stops us.
Oh, and Scott here in the chat room is saying that that grenadier we just saw looked a bit like hollow-snouthed grenadier, so in the genus Sulorhynchus.
So that other identification, the Chlorophthalmidae, must have been the green-eyed fish.
The green-eyed fish.
Which neither of us thought had green eyes.
No.
Go ahead. Thanks. I will ask Joanna. Hold on. So, and this is another one of those urchins that we saw earlier that Chris did not identify, I think. Is that right? And so, just a reminder to you, our biology
we still have
four water samples
to
be taken if we wish.
So I have been waiting a bit to see if we come across an area which would have more fish diversity. But since that hasn't happened so far, I think we're in a good. Since we're getting close to, we're now 520 meters depth. Maybe this is a good place where we could take a water sample.
Why?
Because then we can compare in different bathymetric areas, strata, and the fact that we are not seeing some of the fish here doesn't mean that it's not there. And so actually using these eDNA tools is another way we have to actually look into communities in a given area. And basically this eDNA is the DNA that is shed into the water just as part of excretion from different animals, mucus, scales, skin.
Should we wait till 500 then? If we don't see anything before then?
Yeah, I would say we can wait a little more, and then if we don't find anything until 500 meters, then we should take a water sample.
Okay. Thank you.
Thank you.
I kind of wish that we had taken one at the rock outcrop that you didn't see that had a number of different species. But I didn't feel comfortable doing it on my own. I wanted the biology lead to tell me. Because I'm still learning a lot about the eDNA. It's very interesting how it sort of has a community snapshot, as it were.
Yeah, exactly. Oh.
How many urchins are we up to now? Like 10?
I think we've seen
easily?
Yeah, I think we've seen just such.
And we have not seen this one before on this dive. I'm not sure we've seen this one before at all.
It reminds a bit of Formosoma, but I'm not 100% sure. Because it has these inflated spines at the top. But they're usually much darker.
Yeah,
at least the ones that I remember.
Right, the ones we've seen on earlier dives were much darker. They were like a dark red.
Yes, dark red. And those were like Formosoma placenta, I think, the ones we've seen earlier.
Oh, that's what he's saying this one is too.
Oh, a small Formosoma placenta. Oh, cool. So maybe it has a lighter color when it's juvenile.
That's fascinating. So it could could be a different species, according to Chris.
Okay. And these are very interesting because when you collect them and you get them, on deck, they're actually just a flat disc. They don't look at all like an urchin.
Chris was telling us this, I think, while you were out doing the live interaction, that at least one of these, I'm not sure if it was this one or a different one, it was called a pancake urchin.
Yes,
and that it was the water pressure that kind of held it together. And then when it would come on land, it would immediately flatten out. So he did tell us about that. It was very interesting.
It's kind of sad. I really like the UFO shape.
Yeah.
That's another reason why getting this imagery is so important. Even if we can't physically sample today, the imagery allows us to correlate with samples that have been brought up before.
Yeah.
And as Joanna, you've said on earlier dives, they may be different colors.
Yeah.
Their bodies may be so fragile that it's hard to know what they looked like when they were in situ. And so this kind of work is so very important because it allows us to make those comparisons that we may not have been able to make before.
Indeed. I mean, this Formosoma placenta really puzzled me for a few dives in a previous expedition that I was in, because we kept seeing them with the ROV on the seafloor, and then we were doing some dredging in some areas to collect the fauna, and we were getting all of these pancake urchins, and we're like, strange, they really didn't look like the same thing.
Right.
But then eventually we were able to match them to Formosoma placenta, but I would have never, if I hadn't seen them in the water, I would have never imagined that these two things were the same thing. Just totally different.
And there's another one of those red urchins that Chris Ma had not been able to identify off the top of his head. And this is at least the third one we've seen, if not the fourth.
Yeah, this very dark red test.
Yeah. I mean, so the urchins have been very common. We've now seen a lot of those yellow worms, whose name I can't pronounce.
Terribilid worms.
Yep, terribilid worms. And we're just moving over a fairly sedimented plain. You see little parts where there's hard bottom here and there, but it is pretty heavily sedimented, and I wonder if that's why we're not really seeing a huge abundance of animals.
Yeah, and also not so many corals. We've seen a few, possibly Stichopathus, and earlier on, so a few black corals.
And Bathypathus too,
and Bathypathus, yes.
Yep. We did see one Chrysogorgia. You may have not. You may have been on your interaction talking to...
Possibly.
Yeah. So one Chrysogorgia.
I don't remember seeing that.
It was the only one we saw so far.
Okay.
There was a groundswell of excitement when we saw the Chrysogorgia. We're seeing little isolated rock outcrops.
Yeah. Yeah, they really make these sort of plates, isn't it?
Mm-hmm. It does look very platey.
Oh, and this, I think this dark thing here in the center will be a sponge skeleton, actually. Of one of those fibrous sponges that we usually find in shallower water.
Did you see that little fish?
Mm-hmm. Yes, I think. But it must have been here for a long time then. Or no, it's actually, no, I don't think it's actually a sponge skeleton. I think this looks more like some sort of algae that's come from the surface.
Interesting.
Yeah, some sponges do have like this very fibrous skeleton, and then when all of the organic part is dissolved and rottens, then we see these fibers like that.
The rocks around it look like they have a variety of life.
I think there is probably a bryozoan just here coming into the center. Oh, an anemone that we've seen also earlier on. They have very long bodies.
Should we take a water sample here?
Yeah, that's actually a good idea. We should take a water sample here, now that we're at least a little bit closer to a rocky outcrop. Thank you.
Thank you. Ooh, that fish went quickly.
Yeah, it was a eel.
Didn't go far
of
though.
some sort.
Still
no, it's there at the back.
Yeah.
Oh, that's true. It just got out of the immediate vicinity.
Camera shy.
Look at
that. Oh, how
that.
beautiful.
Yeah, it is.
Is it an eel?
Yeah.
Everyone else says it's an eel. I'm guessing it's an eel.
Yeah. And on the back we can see also a...
I have it. Is it 2?
Yeah, that's number 2.
Did it go?
Yeah, number 2.
Okay.
Great. Okay, and we just got our second water sample of the dive. More urchins there.
So, Joanna, can you explain what we're doing with these water samples?
So sure. So these are intended for eDNA analysis, and eDNA stands for environmental DNA. So basically this is the DNA that we can find in the water column that are a result, so this is what we call extracellular DNA. It's a result, either of the excretion of some organisms, some mucus, cells that they shed, scales, skin cells, etc. And so this DNA floats around in the water, and by collecting this water we can then filter it. So on board it goes through a filtration system, and then we will extract all of the DNA that is in the water, and with it then we using some molecular techniques, and we will amplify short fragments of the DNA. We will be able to make what we call meta-barcoding. So, and it's our small barcodes of DNA that by contrasting it with several databases, genetic databases, we will be able to put a name onto the species. Oh, and we're seeing here Anthomastus. We had not seen these before. These are the first actually on this dive, and actually also on the previous dives we didn't see them.
And actually right before that, when you were telling us about eDNA, we were also looking at a stalked crinoid that was totally out of the sediment.
Outside
it was
of the sediment,
slowly moving along the sediment, but it was not in the sediment at all.
Hey, this is Chris Ma.
Hi, Chris.
Hi, Chris.
Hi, I just wanted to comment a little bit on that stalked crinoid.
Oh.
So those are actually, that's Endoxocrinus. That's actually a crinoid which has a free-living body. The stalk ends in kind of a tail. And so what you're seeing there is the end of the tail has been lost. And one explanation for that is that a Citroid Urchin has predated upon it. This is actually what they do, is the Citroid urchins attack them, and they kind of chomp them up from the bottom up. And so it's not entirely clear what the urchins get out of that because there doesn't seem to be a whole lot of meat or other nutrients. But there is sterium and calcite and whatnot, so perhaps that's what it is. But in any case, it's thought that this might be a very influential part of the ecology on crinoids, and of course a very important part of how we understand urchins in the deep sea as predators rather than as herbivores or detritivores. But given this area, it wouldn't surprise me at all if you guys came across, and they've seen this before, Citroid urchins, or possibly even other urchins, I don't know, but Citroid urchins attacking one of these stock crinoids, because we've already seen another stock crinoid with stock kind of partially removed. So it's possible we could see that, actually happening. So something to look forward to if it comes up.
Great.
Anyway.
Thank you, Chris. That's very interesting.
A very exciting day, so thank you.
And here you were afraid of the...
this is Echinoderm day.
Here you were afraid of the isopod, and urchins are munching on crinoid stems.
I'm totally fine with the urchins. I like the urchins. I like the crinoids. Not a fan of the isopods.
Yeah, and we're having some shoreside scientists saying that if we come across more of those anthomastus, we should have a closer look again.
We will do
to
so
get
if we
some
can.
more, to get some more details.
And that was
of the morphology.
The roundish. It's a coral.
Yeah, it's a coral.
Okay,
yes.
yeah, the roundish-looking coral that the pair we were looking at previously. We'll keep an eye out for those for sure.
Oh, yet another urchin.
Oh,
different and also different one.
And
an eel to the lower right. Yeah. Yeah, it's different, this one, isn't it? Beautiful.
It's very purple.
Yeah, it's beautiful color.
Okay, it's very purple when there's not sediment on top of it. And we did see that eel on the same eel, different eel, on the bottom right while we were looking at this urchin.
Oh, it just went by, actually.
Pilot, this is
and yet another urchin just settling next to it. Look at that. With super, super long spines.
Oh, look at that.
Oh, is that a queen snapper? I think it is. I think it's our first queen snapper.
Is that the queen snapper? What depth?
Yes.
We're at
so it's
500 meters.
499.
499.
Oh, I wonder if this is actually our first. Is that a record for queen snapper? I know there was a record
queen
now.
Snapper. I think that's the first question.
Yeah, it really looked like one.
We have it appears to be a confirmation of a queen snapper in the chat.
Yeah, so we were at 499 meters depth.
Yep, that was one of the species that the local fisheries management council had asked us to look for and identify if possible at particular depths.
Okay, now we really are on a beautiful outcrop. We can see already there some
sponges.
A number of beautiful sponges. And one of them is actually a rock sponge. So the one further to the right that is folded, it's a rock sponge.
It's beautiful.
Yeah.
And the nice thing is when we zoom in on some of the animals here, we'll also get to zoom in on the rock itself. It looks like it's probably a fossilized coral, but it'd be nice to get closer and find out.
Oh, and look at so many things just living on and in it.
And
inside of these crevices.
Oh, wow. That's a lot of shrimp.
Wow. Yeah. Shrimp cave there.
Yeah.
So yes, we have people are in the chat room cheering the appearance of the first queen snapper.
We aim to please. So we're here at the Lang Bank, which is about a 15-kilometer wide bank that's on the east side of St. Croix. And this is our 7th dive, our 3rd in Puerto Rico and the U.S. Virgin Islands area. And what's really interesting is how shallow this bank is. So it's like 15 kilometers long and only about 30 meters depth, 20 to 30 meters depth. And it was formed when it was originally a coral reef that had elkhorn coral and another coral and sort of massive corals on top of it. And what we're really seeing right now is probably a piece of limestone that looks like it might be fossilized coral. It may not be. It wasn't quite as clear as we had seen earlier. But you can also see that all of the rocks in this area are composed of a mix of coral and sand and sediment rubble. So it's really very interesting. And this is a standalone little outcrop here at Lang Bank.
And we're starting to see here a number of sponges that we've been looking for earlier. Oh, and actually also corals there to the left. But let's have a zoom in on this sponge here.
That's a sponge?
Yes, it is a sponge. You can certainly see, all of these spicules protruding from the surface.
Now I see it.
So, and it is a glass sponge, possibly in the family Roselidae. It seems to be broken for some reason, the surface there. And you can see how the sediment accumulates on these spicules at the surface, and that actually protects it from clogging, because otherwise the sediment would go into the sponge through those small openings that it has all along its surface.
Mm-hmm. Wow, just look at this rock, though. I gotta say,
it's amazing.
Super cool.
Yes. So we actually have.
It's a habitat for so many things. What is that we're zooming in on?
I think this is again a black coral. It's just a different one that we haven't seen this before on this dive, or actually also not on our previous dive. And there's a shrimp just sitting on it. Let's see if we can have a zoom in on the black coral to see in greater detail. Okay, so it was the maximum zoom we could do now. Oh, there's another one.
And I'll just note, this is not a very big outcrop. If you look in camera 2, where you can see the view from Sirius to the Deep Discoverer ROV.
It's just a block.
It's a block that's about the same size as the ROV, and yet look at how covered it is. So it has this, all of a sudden you've given a hard substrate to the animals on the seafloor, and they're taking full advantage of it.
And so it seems that the coral that we were looking at earlier on was possibly Antipathus. We're getting an identification here in the chat room. Oh, there's again one of those Olopus crinoids, and this is the same species that we were looking at earlier on.
Oh, the crinoid! It's
look
so
at that
cool.
To the left.
And this one's a little more open.
This one is a little bit open, yeah.
Mm-hmm.
So there was actually a species, I'm not sure it would be the same species that was described from the Caribbean not long ago, and that was Olopus mikihe.
So there was a request to look at the long white stalk to the right of the sponge.
Long white stalk to the right of
I'm not sure which sponge.
Yeah. That's probably that.
The long white stalk
I guess
to the right of the sponge.
Yeah, I guess we know what it is now.
We got there eventually.
We have another fish there to the left. Let's have a look at it.
It's trying to hide. It's beautiful.
Look at that. I wonder if it's also some kind of snapper.
I don't know, but I think the rock looks quite fossiliferous. Do you see all the structure that's in the rock itself? So I'm guessing it is a fossil coral of some type. Oh, what a beautiful fish. All right, we should eventually try to get to that white stalk at the top.
Yeah.
What's in the top left? That
was a bivalve.
It looked like a bivalve.
It looked like a clam file, maybe. I don't know. I couldn't see.
Wow. What is that?
What? That's a really good question. What is that?
That's really cool.
So
looks
colorful.
Like it looks some sort of bivalve.
I would agree with that. I can see the two shells.
Yeah, but
is that coming out of the bivalve, or is it something else?
Something living inside the
Phyloclam.
bivalve shell.
That's what I'm thinking.
So they're calling it. Scott Friend's calling it
that
a
was
phyloclam.
that was my first guess as well.
A cesta?
Yeah. In the Northeast Atlantic, we have or the Azores and Mid-Atlantic ridge, we have one species that Cesta excavata, but it looks a bit different from this one.
This one's very pretty.
So like an orangey. Oh, and that's again one of those corals, black corals here.
I think that's the same one
very
we zoomed
thin.
in on.
Yeah.
What a cool find.
Yeah. And you can, this is it's a very good example. As soon as you find a good rocky substrate, how much more diversity you can find associated with it. This could actually be a good spot to have also
another water sample.
Another water sample.
Okay. Now this is watch lead. Would we be able to get another water sample here? Thank you.
Thank you. Yeah, we wanted to zoom in also on that stalk there. Once, yeah. Great, thank you. And here we see again one of these glass sponges with one crinoid on top of it. Oh, and a little brittle star also living here associated to this black coral that has these yellow tentacles. And yet another glass sponge, different species here. And this actually we have seen earlier on, and these really bright white dots that we were already wondering about it the first time we saw it. I wonder if it are indeed zoanthids that live inside of the sponge. So this is relatively common to see some of these glass sponges with zoanthids living on the inside in the canals systems. Oh wow, look at this rock, it's amazing. Also a pompon anemone there, Liponema. Another one of those very tall anemones that we've seen earlier. Let's have a zoom in also. Can we have a zoom in on this whip that is just center on the screen? And again, this has a brittle star associated with it, as we've seen earlier. Only the color on this whip coral here is different. So the ones we've seen earlier on, they were orange, and this one is rather pale white. However, the brittle star seems actually similar to the one we saw earlier. So yeah, so perhaps a Stichopathus of some sort. Oh.
I do love the name Stichopathus.
Yeah.
Because they look like sticks.
Yeah.
It works out for me.
Yeah, and so apparently the yellow one we saw just to the left of the other sponge, or it's actually showing here now, to the top, might be this Stichopathus. So another genus. Oh wow, so much diversity just on this one rock. It's quite amazing.
That really makes sense though, right?
Yeah.
Because we haven't seen that many rocks. We haven't seen that much hard bottom, so they're all going to congregate in a place where they can thrive. Right?
Ooh. And what is that just underneath it to the right? Let's wait to get this sediment settled.
And what's the pom-pom anemone on?
I think it's sitting on a skeleton of a dead sponge.
That's what I thought too.
Yes. Yes.
See, we can keep playing sponge or rock.
Yeah. Yeah, that looks like the skeleton of a sponge that was living here, possibly a long time ago. From the structure of it, I would say it was a glass sponge. And glass sponges, some groups have this fused skeleton, so they are hypersilicified and have this fused skeleton made of silica, which makes them to not disaggregate completely when they die. And so it serves also as a very good hard substrate for other organisms to live on.
Yeah, it's really interesting to me that it's against the rock, and then the pom-pom anemone is on the sponge rather than being on the rock itself.
Yeah.
Have we imaged everything we need to image?
I think there was still... okay, let's have a closer look here at this yellow black coral. So this is the one that our coral experts here in the chat room are identifying as the Stichopathus.
And pilots, if there's any way that we can zoom in on the white stocky thing that was near the top, near the sponges, there's been a request from the chat room. Yeah, that's about the best I can do. Keep going up, and I think it was over to the... Oh, well, there is one to the right, but that's not the one we were talking about. I thought it was up to the left, but maybe I'm wrong.
There was one further up to the left, I think.
But look, are there two more crinoids up on the side?
Three of them.
They're like the fist?
The holopus, yes. Yeah. And we were right to think when we saw them the first time that they really look like prehistoric, because apparently they have existed since the early Jurassic. So there are 6 species within this Holopodidae family. 3 of them belong to the genus Holopus and 3 genus Cyathidium. And so when when they close their arms, they really make this sort of a completely tight fist.
Right. Compared to the crinoids that we often see, which look much more fluid and
and open and
open.
Sure.
All right.
Not with those robust.
So here's one of the 2 remaining
arms.
whip corals.
Yeah.
The other one is on the far side.
Yep. Oh, and the one actually, what I was pointing at, that's again another pompon anemone, and this is much more the color. Is it? Although it's not moving nearly as much as the other one. So this is much more the color that that we typically see them on. So we've now seen this, sort of a brownish red. If we could just zoom in on this whip coral.
Then I think we'll be done.
Yeah, then I think we're good.
maximum zoom.
Yeah, we can see there the base attached like with a sort of a disc to the substrate. Yeah, and it seems to have like sort of rather short tentacles there. But we can see the dark skeleton underneath the living tissue. Yeah, thank you.
Shall we move on? See what there is to see
I feel like saying no, but just because this outcrop is so cool.
It is.
We're seeing lots of different things here. But yes, we should definitely continue.
Yeah.
We have to find the next amazing outcrop of biodiversity.
Exactly.
Hey, I suggested that we move. No, I do think it's important to keep moving up slope, because what we are trying to look at is a transect from the lower depths to higher depths, and we want to be able to see what might change as we go. So it's fun to get stuck at these beautiful outcrops, but
hopefully we will find some more of these along the way.
We also expect it to get steeper as we go further up, which could lead to a really interesting transition in species diversity.
What is there we're seeing in front of us? Looks like something very angular, possibly man-made.
possibly a rock.
Yeah.
We'll find out when we get there. Oh, look at that. So this is, I think, I forgot to write down the name of it. I think this is the sea urchin that we've been seeing earlier, which may be dead with a brittle star on top of it. I'm making a wild guess that that sea urchin is no longer alive. I may be wrong. But it doesn't appear to have its spines anymore.
Yep.
Conolampus, is that what it was called? Does anyone remember?
There have been a variety of urchins, but that is one that we were looking for.
I think that I'm going to take the wild guess that that was a Conolampus, and that it was, or something very close to that in wording, and that it was no longer alive. And we'll see if I am right. And so this has already been a truly very interesting and exciting dive. We're here off the coast of St. Croix. We're about 9.5 miles away from the coast itself. But there's this very long feature that we're on called Lang Bank. And it's incredibly important for fisheries, and that's one of the reasons that we're here looking here. It's a very, very shallow bank. We are actually climbing up the northern slope, heading towards the top of the operational area that we can get to. That is definitely not a rock. Definitely not a rock.
Can confirm.
Can confirm. Oh, look at that. The brittle star is on the move.
I think the brittle star sees this piece of
trash can?
Human detritus as a good perch, good place to get nice and high up into the water column.
Mm-hmm. And that one only has 4 arms. That
That'll
doesn't
happen.
seem Yeah, I was going to say that doesn't seem quite right. Although in one of our previous dives, we found out that not this particular type of echinoderm, but some of them actually only do have 4 arms originally. It's quite beautiful.
It's a great zoom. Thank you.
And I think what's been so interesting about this dive, although I keep hoping that we're going to see more fish, has been this occurrence of these big dead sponges that we know are coming from somewhere else, but they're not where we are. So this is just a really fascinating dive. This is also an area that's really great to think about biological connectivity across the entire Caribbean and Atlantic. And so that's one of the goals of Aspire, is to understand how the biology may connect from one site to another. And so I think that that will be really helpful with the samples that we image or collect. And a lot of the work that we're doing here builds on previous work that has been done around Puerto Rico by the Okeanos Explorer and others. There have been at least 2 previous expeditions to this area in 2015 and again in 2018. Plus there's been some mapping done earlier this year. And so we're really privileged to get to come back here and explore even more of the areas around Puerto Rico and the U.S. Virgin Islands in the EZ. So it's really great that we're able to add to the body of knowledge that has already been collected here.
And really complement also the work that has been done in the shallower portions of the Lang Bank by researchers and research administrations here in the area.
And they've actually been out this past week with a smaller shallower ROV.
Yeah.
Yep.
I think they've been mostly serving between or down to 130 meters depth, so the fact that we can dive considerably deeper really adds on to that. So we can really complement their work.
So what kind of coral is this, Joanna?
I would say it's again a black coral, possibly in the Stichopathus genus.
That would have been my guess, but it would have been a guess.
And again, it has the brittle star associated with it. So this has been a constant throughout this dive. Whenever we've been seeing these Stichopathus corals, they always have one of these brittle stars associated.
Pilot, when we have a chance, could we zoom down to the base of this coral? So one of the scientists in the chat room, Jeremy Horowitz, is interested to see what this might be attached to. Oh, and it's really hard to tell.
Please move it,
because it's hiding.
I don't think they move that quickly.
No.
But some of them do move. Is this one of the kinds that moves?
No, I don't think so.
No?
No.
Okay.
And there's a shell up to the top left.
That's a large shell, gastropod shell there.
All right, we've made it to 480 meters. Our goal is to try to get to 300. So we probably should continue to move on just a little bit.
We're
getting okay though, right? Yep.
ROV nav says we're on track for that.
Excellent. So. And so then the temperature is 13.7 degrees Celsius. So it has been getting warmer as we've been going up, which is not surprising at all.
Not at all. And it's also interesting to see because we've been trying to escape the sargassum patches for the past couple of days that have been preventing us from diving. And it's also interesting to see that in the previous dives where there were more sargassum at the surface, we could also find some of that sargassum on the seafloor. And here we haven't been seeing so much of the sargassum on the seafloor.
It really has been sort of the bane of our existence for the last couple of days. Unfortunately,
it really has.
We lost dives for the last 2 days simply because there was so much sargassum, not just at the surface, but also about 15 feet down, which is exactly where our ship has its intakes for cooling the engine system. And the strainers that keep the weird stuff out have been getting clogged with sargassum. So it's been a little bit of an operational challenge at this time in and around Puerto Rico and the U.S. Virgin Islands.
Yeah. So for some reason, it seems that you're in an area where we're finding less of that sargassum, which has been great because it really enabled us to dive.
I should mention, though, that the engineers have been doing an amazing job.
Oh, yes.
It's been very hard, for them to make sure that we are safely operating, and that's a really important thing. So I actually do want to give a shout-out to all the engineers here on the Okeanos Explorer who've been working very, very hard, not just every day, but the last 2 days have been special.
Yeah.
What is that?
You go, Deb. Rock or sponge?
Not a rock.
Let's have a look. Oh, and actually, if it is a sponge, it's a dead sponge, and it looks like it has like sort of a bacterial mat on it. Oh, and this is very interesting. So this is a Citroid Urchin, and it has one of those sort of spider crabs on it, or a small king crab on it. From a distance, it looked like just some extra spines on the urchin.
I was going to say, it looks like another urchin sitting on top of an urchin, but no. So that is a sponge. It's just...
Yeah, it's just dead, very much dead. And you can actually see because the surface is slightly broken there on the top right, so you can look a little bit inside. But for some reason, so it seems to have created this bacterial, this white veil on top of it. It's looks like a bacterial mat that is growing on it.
That is really interesting. We haven't seen that yet on this dive.
And that's probably because we've been seeing mostly already the sponge skeletons that completely exposed, which means that they've been there for much longer time. So this is probably dead not that long ago. So it's still undergoing this rotting process, and it's not uncommon then to see some of these bacterial mats growing over them. Is that one of those urchins? Again, possibly there at the lower right, but seem dead.
I don't know. It's amazing to me that, from the distance we are right now, you think, oh, there's not much on the seafloor. And then as soon as you zoom in, there's so much to see.
Yeah, and we shouldn't forget of all the small, infauna that lives in the sediment as well. But that largely goes unnoticed unless we do some sediment cores and bring those into the lab, and sieve all that sediment and look at all the diversity that lives within it.
So, Joanna, what's been your favorite part of this dive so far?
Oh, that big
just
block.
Dive 7.
That big block that we've just came across. Oh, there's actually a bit of sargassum here, see? We're talking about it, and there he goes.
Maybe we shouldn't talk about it.
No. So my favorite part so far was really that big carbonate block that we just came across that was just, covered in so many different groups and so many different species. That's really spectacular.
I think I like the jar full of hermit crabs.
Oh,
so
I did not see that.
You will love it when you see it later.
was in that live interaction.
Yep.
Yeah, again, this looks like a sponge skeleton here. a much more degraded one. And I think there's another coral here coming into view. Let's have a look. And it looks different from what we've been seeing so far, because this one is branching. It does have, again, this brittle star associated with it, but it looks a different morphology, so it's not the Stichopathus that we've seen earlier. Also, the brittle star actually looks different.
I was thinking the same thing.
Yes, it has these stripes all along its arms.
So one of our scientists in the chat room mentions
Yes,
that's a
it's
soft
a soft
coral.
coral, so a different group.
Plexaurid.
Plexaurid. Oh, look at how it's uncoiling its arm. Look at that.
That brittle star is so
look
cool.
at that, yeah.
Because it's so stripy.
And there's something else actually there with some sort of a crab there.
It has very long
with
legs.
Very long legs.
Could it be a sea spider? I don't really know
No,
what those are.
No, it
They
doesn't
just
look
weird
like.
me out.
No, it doesn't look like. So, yeah, it's some sort of a squat lobster, but I've never seen them with these long arms like that.
So we're getting an identification
it's really,
of an
really
asteroschema for the brittle star with the stripes.
Oh,
called the snake star.
asteroschema. Okay.
And a chirostylid
Oh, the
crab.
snake star. That's a chirostylid crab.
And then perhaps a dendrobranchia.
Branchia.
Yeah. So that was a good find.
Asteroschema. The snake star, yeah, it really looked a little bit like those arms. And with those rings.
Very stripy.
Yeah.
And as we've mentioned earlier, if you're just tuning in, though, you might not have heard that this is actually a very highly bioturbated area, we think. I think there's a lot of things that are living just below the sediment and that are burrowing into the sediment or popping out of the sediment. So we're seeing a lot of interesting traces along the seafloor as we move.
And we're now at 465 meters depth, so I think seeing that queen snapper was really a treat for us at nearly 500 meters depth. Oh, and we have here again the same coral with the same brittle star we just saw earlier, just a while ago. It's really pretty.
That is amazing. So a snake star, huh? Might be my new favorite.
And as we go through our dives, it really makes me think a little bit about the parallels with what we've been finding earlier on this expedition up in the Azores Plateau and the Mid-Atlantic Ridge, because this is actually a very interesting area where in such a short distance from from shore. You have all sorts of deep sea ecosystems, you know, from tropical reefs to canyons, seamounts, and even what's the deepest trench, oceanic trench that there is in the Atlantic here, the Puerto Rico Trench. Yep. And this really promotes a lot of biodiversity. And while we have seen, you know, different species and some different groups, we have already seen some species that are shared also with the Azores Plateau and the North and the Mid-Atlantic Ridge.
And as you said, that's one of the goals of the Aspire campaign.
Exactly, to look at the connectivity.
Look at the connectivity, and then also to keep looking at unexplored areas that we haven't been to before.
Exactly. So there was, for instance, the six-gill shark that we know also occurs in that area. There's been also a couple of glass sponge species that we found in previous dives here that are also known from the Mid-Atlantic Ridge and the Azores Plateau. So this actually gives us a great opportunity to expand a bit, you know, the geographical scale at which we look at these distribution and diversity patterns. And it's quite common that when we're focusing on species that occur in the Mid-Atlantic Ridge, we tend to compare a lot with the fauna from the European margin. But also now seeing what we've been finding on these dives really puts it in a larger perspective, you know, an all Atlantic, all North Atlantic perspective. So at least for some of the samples that we've collected previously, it will be great to examine them, and also from a molecular perspective, look at, you know, how closely related they are in terms of population, at the population level. How closely related are they to the populations we may find on the Azores Plateau and on the Mid-Atlantic Ridge?
And that's one of the reasons that we don't just collect things that are rare fauna.
Exactly.
We often collect things that are actually characteristic of the site, or sometimes we collect things just because we know or think that there may be an opportunity for connectivity.
Exactly.
I'll note that we're not sampling on this particular dive today because we are within the 12 nautical mile limit, but we are really grabbing some fantastic imagery that's still going to help with those goals. Well, what's crawling at the top left? Did you see that?
Oh, no, I couldn't see it.
So is that a cup coral to the bottom?
Yeah, that's a cup coral. Yeah, I don't think it's live anymore.
Right.
You can only see the septa there, the calcareous.
And we're looking down at the stalk?
Yeah, we're looking at the stalk. And so for those that might be joining us just now, we are here on our seventh dive of this EX2206 Voyage to the Reach campaign. And today we are characterizing the Lang Bank. So this is a large bank located just 9 nautical miles to the northeast of St. Croix Island. And we're doing our dive ascending the north slope of the Lang Bank, roughly between 750 and 300 meters depth. This area is particularly important as a spawning area for a number of commercial fish species, especially in the shallowest portions of the bank. And what we're doing is trying to characterize the habitat and the benthic and fish communities that occur in the deeper portions of the bank. So we've already seen a number of fish species, but also corals and sponges, certainly a large diversity of echinoderms, both sea stars, brittle stars, sea urchins, and crinoids, which are actually now showing here in front of us.
Stalked crinoid?
Yes, a stalked crinoid.
What's to the left of the stalked crinoid?
Let's have a look there.
So we've seen two like this before.
Yes, so these are the ones that Chris was referring to as, you know, being predated upon by Citroid urchins. There is not one in sight, so he's safe for now.
What I find amazing is how long sometimes these predations take.
Yeah.
On one of our earlier dives, I believe a cookie star was eating a sponge, and the idea was it could have been doing that for months at a time.
Weeks or months. Yeah, and Chris and I have been exchanging some messages about that, because sponges also have the ability to restore, regenerate lost parts. So we had been discussing, throwing around this idea that that could be a supply of food forever for some of these urchins, if the speed at which the sponge can regenerate its tissue is higher than that it gets consumed. So you would have like a continuous supply to the urchin or to the sea star. It's just a wild idea. But you never know. Deb, it seems that we missed a queen snapper going by.
A queen snapper? I saw a silvery fish.
Okay.
But I did not see what I thought was a queen snapper, but maybe we just didn't see it. And here's another one of these that might be the snake star again.
The snake.
Love it.
It's beautiful.
So we could take some time
getting
looking at this nice coral and brittle star combo.
So now we've been seeing quite a number of these. And to the left it looked like what was an argonaut or an argonaut shell.
What's an argonaut?
Yeah, it's this mollusk that builds... it's actually, it's not a mollusk, a cephalopod that actually builds this paper-thin shell, and it's really beautiful, super delicate. And I think we just saw a broken shell there on the seafloor.
Very cool. Oh, so the queen snapper was in the distance outside of the full range of the lights.
So we did not see that.
We were talking. We apologize.
Yeah, there is another coral here. See if we can have a quick look at it, or sometimes they're so difficult just to see against this
against the carbonate, certainly. Yeah.
Especially if they're not like bright colored.
So far I'm a little surprised. I really thought we would have seen more fish by this time.
Ooh.
Extreme close up.
This is different. I wonder if this is a chrysogorgid coral. I understand that you guys had seen one while I was away on my live interaction, but so I'm not sure if it was the same thing or not.
I'm not sure. So they did think that was a chrysogorgid.
Oh, it was. Mm-hmm.
Oh, there's another little sea star.
This really has been the Echinoderm dive.
Yeah, it really has.
Incredible diversity that we've been finding here.
That one's cute, and it's got its arms up in the in the air. Well, in the water.
Wonder if this is one of those goniasterid sea stars.
It has very long tube feet.
Yeah, it does. I always find it so interesting to look at how they, coordinate all those tube feet to move.
Mm-hmm. So that was a relatively small sea star compared to what we've seen on this dive so far.
Yeah, we've seen one that was like 40 centimeters long.
Hey.
Hi, Chris.
This is Chris Ma again. Sorry. So this is, you're correct. This is a goniasterid. This is called Anthinoides pierci. And one of the reasons I called in is mainly because I've been studying this animal for years. There are multiple species in the Pacific. And I don't think I've ever seen a live one on the Atlantic side.
Oh, wow.
I don't think I've ever seen a live one on the Pacific side either. So, I'm really I think there were a couple of pictures from early Johnson Sea Link dives, but this is the first time I've ever actually seen one, actually in situ myself. Obviously I'm on the ship, and I'm seeing it with everybody else, but I'm just overjoyed to actually see one alive, and to see what color it is, which is another, for me, quite a. There's a whole bunch of, I don't know what you call them, classic species. You know how when you're enmeshed in something, and there's everybody says, there's all these species, and this is the classic fauna of this area, so you have to at least see one of these. And so I've been working with the tropical Atlantic sea star fauna ever since I got to the Smithsonian, but even before that. And so this is the things that we've been seeing today are some of the classics, the things that are, if you study Atlantic echinoderms, some of these species are among this is what characterizes the Atlantic fauna. So Nymphaster Erinatus, Anthinoides pierci, Coronaster briareus, Conolampus, these were all things that I used to know only from plates in a book and dead specimens and and things like that. And I'm learning so much about the brittle stars, sometimes frustratingly so. For example, that Astroschema, that snake star. There are, I think, 3 species known from the Puerto Rico area.
Okay.
And, they're difficult, to say the least.
Okay.
They're really hard to tell apart because of various factors. So, anyway, I just wanted to call in and just proclaim my love for Anthinoides pierci.
Excellent.
And it's been a banner day for echinoderms.
Excellent.
So thanks so much.
Thank you. Really happy to be able to bring this joy to you. And to us.
Even if we're not collecting it.
And to
it's
us.
kind of weird. Yeah. Well, it's kind of weird, because I almost don't want to collect any of these because we have representatives of all of these species in the collection already and from Puerto Rico. But the video footage of seeing them alive is just priceless.
Yeah, it really adds on
it's amazing,
a lot.
A valuable thing for me.
Brilliant.
So, I just... so I'm very thankful and to everybody, and I'm glad that we're in the water today. So, and for this is just an incidental too. I've been pursuing that snail that was seen on that stalked crinoid with the proboscis over the anus, and it turns out that the snail is quite rare. We know what it is. But it is quite rare, and we're tracking it down, and the video footage might be surprisingly novel. So
that's excellent.
Lots of fun discoveries today. Yeah,
right.
Yeah, I'll send more as I know more for sure. Anyway, thank you.
Excellent. Thank you, Chris. Thank you so much
for
Chris. Sharing the knowledge with all of us and with all of those that are following us.
Very
all over the world.
Thank you. Bye-bye.
Well, we just found out that tonight's entertainment is the Caribbean Trivia Night.
Oh, okay.
Ooh, look at this little fish.
Oh, look at this little fish.
And the little crab.
And the little crab there in the back.
I'm going to guess that this is not a commercial species.
No, or perhaps a juvenile.
Yeah.
If any. Oh, actually, it looks like a very small slime head. Could that be?
Could it be?
It sort of has like this slime head look to it. Could this be a juvenile slime head?
That is what Michelle is saying in the chat room.
Is she?
Mm-hmm.
Oh, yes, it is. Mm-hmm. It's just that the mouth and the eye looks a little grumpy.
It does look a little grumpy. But also, look at the sediment that it's in front of. It's so, it's amazing. You can really see that the coral rubble and all that biogenic sediment that is behind the fish.
And so seeing these steps is actually very interesting because sometimes several species actually have their nursing areas and spawning areas in deeper water, and then the juveniles are, sort of reared there in in deeper areas, and then the adults move further up in the water column.
And so we're just taking a close zoom at this rock,
which has a beautiful encrusting sponge, or several. Yeah, and we can see actually on this sponge the canal system through which the water flows. So that was a juvenile slime head.
Very good.
Really good. And it's way more silvery as a juvenile than it is as an adult. It's usually this darker red color.
I like how we're just seeing these isolated little rocky outcrops. Sort of there's a rock here and a rock there. Most of the time we see something living on them. It's quite interesting.
We did come across a while ago, I think, what seemed to be a fishing line.
Mm-hmm. And we were just talking about that in the chat, that generally when we see fishing line, there's a danger of entanglement for the ROV. So we moved away from that as quickly as we possibly could. We don't really hang out to zoom in on them or look at them unless there's a reason. And so the idea was to try to get out of the area where the fishing line was. Here's another small rock.
Yeah, and so this shows us, that we should also look under these small rocks. Sometimes the small juvenile fish will actually seek shelter there. So perhaps we can see some more of those.
We've got a nice sea urchin to the bottom left. Am I looking?
And some more of these
it's walking.
crinoids. Oh yeah, the urchin is walking.
Yeah.
Yeah. So that is the one that you were talking about earlier on, that some of the spines look like they were Q-tips, right? Look at that.
I'm not sure.
No?
I am not sure.
Yet a different one.
Yeah, I don't know. I would not swear that is the same one that we saw that had the Q-tip-like spines.
I have to say that the echinoderm diversity here is just astounding. Oh, look at that.
This really does not look like the one we saw before.
Okay.
No.
And it's also not chromosoma placenta. So yet another one. Maybe Chris will call in.
Or at least tell us in the chat room what it is. And, if we can zoom in as close as we can underneath any of the rock faces looking for juvenile fish, that would be an amazing thing, because it would certainly help to understand, as you were saying, whether this is a place where juveniles might hang out.
Yeah. Oh, that's great. Thank you, pilots.
Makes me think of peacock feathers.
Yeah. And so this one is roughly roughly 10 centimeters long.
So it's another Echinotherid, which we did see before.
Perhaps Areosoma.
Which I think is different than the one that we had seen earlier.
There we've got some of those bright orange shrimp. A few scleractinian corals as well. Some of those cup corals.
And so now maybe we'll zoom in a little bit on those crinoids. What's next to the stalked crinoid on the right?
I think it's a sponge. And this could be, it seems from a distance, it could be actually one of those rock sponges that I've talked earlier. Actually, the Caribbean Sea or the Western Tropical Atlantic is a hotspot diversity for those rock sponges. So these are within the class Themospongia. They're distributed in a number of families, but they have this hypersilicified skeleton. So it's they're called rock sponges or lithistid sponges because they have these special types of spicules called desmas, and that are articulated and super sturdy spicules and fused. Yeah, and I think this is one of those rock sponges. So that makes their consistency to be rock hard.
This is just a beautiful shot of this crinoid.
Yeah, it is amazing. So unlike other sponges, which are much softer in texture, if we would try to possibly collect this one, it would prove challenging because they can be quite hard.
But the nice thing about these is that, if I'm correct, even if you were only to take a small portion of it, it would regrow, it would regenerate.
Yes, in most cases, yes. So if we don't interfere with the basal portion of the sponge that it uses to attach itself to the sediment or to the substrate, in this case, if we remove a small portion, it will regenerate. However, with some of these, actually, we need to get the complete specimen because often there is like a differentiation of the skeleton composition in different portions of the sponge. So sometimes we may have to look both at the the apical surface, but also at the basal surface.
All right, in search of more small or large rock outcrops.
Preferably large.
Preferably large. That would allow us to look for some juvenile fish. What's that on the right?
Yeah. Probably something that came from the surface.
I don't know. I'm not going to speculate on that yet. It does look like
a root of some sort. Yeah. A bush of some sort. I wonder if it could be actually be also some sort of a coral skeleton, but it's very degraded now. It looks like it has been here for a long time, and actually we see some more of those tubular sponges that we've also seen on the sediment earlier on. Oh, someone here in the chat room is calling the sponge a skull sponge.
I also thought it looked like a skull, but I wasn't going to say anything. On one of our dives a couple of days ago, I kept seeing faces. Every time I turned around, everything had a face.
Yeah, and this is again one of those rock sponges, only here it has this cup shape, with some of them attaining over 400 years of age, which is quite a lot.
I don't know if
There's
that
something
orange thing is.
yeah. Urchin? Yes, urchin.
Why, yes.
Oh, wow.
What is that?
But I'm not sure it's...
Hang on, we'll try to get back to it in a minute. What is the genus of the
There are several,
sponges?
actually. So several families and several genera.
And those are the
I can look
Nymphaster
at-
sponges?
Yes.
Okay, so now
So
we're
it's an informal
investigating.
group.
Yeah,
Yeah,
so now we're
let's
investigating this
have a look at this
orangish
urchin.
thing that we think is an urchin. Hope someone's keeping account of all the different urchins that there are.
Oh, wow.
We have not seen this one before.
No. Oh, and it is alive. And it's carrying quite a bit of debris on top of it.
Yeah.
Of himself.
I wonder why.
Like, I would say camouflage, but we always use that as a as a possibility, right?
I think he just doesn't have any arms to wipe it off.
This is very interesting.
Yeah.
Hi,
Hello.
Chris.
Hi, Chris.
So I am going to correct both of you because we have seen this multiple times today, but never a live one.
Oh.
This one, and I'm going to correct myself about the prior three that we saw, but that was a definite living Conolampus. And,
Oh, okay.
so did you know, I don't know if you noticed it, but if we were to look at that again, there's actually a track right behind it. And so that's the kind of thing that's reminiscent of Conolampus when it's alive. It leaves this meandering kind of maze-like trail behind it in the sediment. The sediment, the gooch stuff that's on the top of it, I don't know what that is, but it's not unusual for urchins to have what's called a collector's response or collecting behavior. Typically, we tend to see it only in shallow urchins, which they collect materials on the spines to protect against UV light. But down here, that's obviously not as much of an issue. So it could be to conceal from a smelling point of view from predators, or it could just be material that accumulates on the surface.
That's great.
And they just kind of let them build up incidentally.
Thanks, Chris.
So we finally found the living one.
Yeah.
Yep.
That's excellent.
We're going to turn now to the rock we just found because we are expecting to see a lot here. But thanks, Chris.
Yeah, and we're watching what we're seeing here now.
I'm seeing another urchin.
And we're seeing here now a glass sponge. I think we've seen this species before. And a roughy.
Yeah, there's several fish that are in this.
right
so
underneath.
pilots, I think it would be great if we could look for all the fish.
Yeah. Let's have a look at the fish first.
I think that would be a wonderful idea.
Yeah. I think we will want.
I think we're going to try to image all the things that we see at this rock because we are already seeing a high level of diversity. We are seeing the crinoids, we're seeing sponges, but most of all, we've seen at least 4 fish so far. So we do want to image them as possible.
So this one is that we're seeing now here to the lower right is the adult, possibly an adult of the species we saw earlier as a juvenile. So that's the Darwin slimehead. And there's also these, smaller fish. These ones I'm not quite sure what they may be, but there's a number of them. So possibly, again, juveniles of another species. But I think we've seen earlier also some other silvery one. I think there's actually one there further back. Oh, and an hermit crab going for a walk.
So that's the slime head, or is it the roughy again?
Yeah, that's the roughy slime head.
Yep. And those are the same
the Darwin slime head. This one.
Take a look at the rocks though, while we're also looking at that fish.
There's plenty of those olopus.
Yeah. And no, I actually meant the rock itself. It's filled with fossils, and it's such an interesting shape. It's really weird looking.
Yeah, it really is.
But every time we zoom in on the rock, you can see fossil coral, and it's really quite interesting.
Yeah, it has a very unusual shape, though.
Yes,
this rock.
It's weird. Oh, I wonder what that tiny little fish is that just went by. So interesting.
Yeah. So would this be like a fragment of that ancient reef that occurs further upslope that would have been dragged here?
It certainly could have fallen down the slope, but the shape is really odd.
It's odd. It really is. And here actually we can see a number of those, prehistoric looking crinoids, the holopus, in various, and they still have actually arms open here.
Yeah, they're all over this rock.
So it seems that they do like this sort of substrate, and they cement themselves to the substrate, so they do not have a true stalk.
Yep.
Oh, look at that. It's actually opening.
That is
amazing. Of them, that's amazing footage. There's really something. They just really look prehistoric. It's incredible.
Ooh, look at the little tiny bluefish. Yeah, those crinoids are amazing. They really do look like living fossils compared to so many of the other crinoids that we've seen. And then the rocks are that's all fossiliferous limestone.
Yeah,
it's super interesting.
Oh, and I think we have here also some stilastrid corals, so some hydrocorals to the right. These white branching.
And it looks like we have an entire school of these little tiny fish.
There are many of those.
Yeah. Interesting place to hide.
Not hiding a lot, but
no,
just
did not work.
No. So yeah, I'm wondering if these are, juveniles of other species that can grow much larger.
Hopefully we will get an answer. We're going to stay here and sort of work our way around this rock and look a little bit different things.
Fish species.
Because there's so many things to see here.
Let's have a look there. Oh, that's a funny. It's a funny shrimp. Almost looks like it has a horn.
And so while we're looking at all these amazing things on this outcrop, I think we have about 2 more hours left in our dive, and only about 100 meters to go. So we've been making really excellent progress thanks to our fantastic ROV pilots. And so we've really had a chance to stop and look and enjoy, but still, keep making the progress that we need to make. So it's been really fantastic. This is just amazing with all these crinoids on it.
And those cryptelia, possibly, those hydrocorals.
I wonder if this rock was once subaerially exposed. I wonder if it was once above, above the waterline. It's such a strange shape.
Yeah. There's another fish here coming into view. There's also a large anemone there to the bottom right. Wow. When we look at it like this, they almost look like giant whale bones or something, isn't it? They have like this odd shape.
Mm-hmm. They really do have very odd shape.
Science got a good aerial view from Sirius Stream 2.
Oh, look at so many small fish there at the center.
Oh, yeah. And for those of you who may be excited by fish, but not only entirely excited by fish, camera 2, the view from Sirius that shows the morphology of the boulder structure that we're looking at is also really cool. But that is a lot of fish. I wish we knew what kinds of fish they were. Pilot, this is Watch Lead. I know it may not be possible, but if you have a chance to zoom in on the little fish in the center, that would be great. Ooh, what is that?
That's some sort of crab there. At the top, it's difficult to see it. We're seeing it from underneath.
He's living in the penthouse.
But you can see all of these hydrocorals, these white branched corals that we just saw.
We're going to try to take a look into that
look
bowl,
at the fish. Yeah.
if we can. It's a bit of an awkward angle, I think.
Yeah.
But the goal is to try to ID these fish if we can, and so getting as close to them as possible. It'll be helpful to be able to zoom in as much as we can and maybe potentially get an ID. Oh, wow, look at that zoom.
Yeah, wow. Yeah, and we see also some encrusting sponges and a small glass sponge there. But let's try to focus a little bit now on the fish.
They all kind of look like they're the same.
Yeah, and these are really tiny.
Could we put the lasers on? Or are they already on?
They might be even on. These are probably, I don't know, 3 centimeters or so.
There you go. There are the lasers. So
yeah, they're really
you're
small.
right. They're 3 to 4 centimeters.
Yeah,
really tiny. Just when you think you get a good shot of these little ones, they hide behind the boulder.
Yeah, but you see just how zoomed in we were once you see when our videographers just zoom out. Oh, look at interesting.
What are we looking at?
The fish, how they were
circling?
Circling, like in a position. That's very interesting. Oh, and some of them are probably just 2 centimeters. One. Look at that.
Do we want to take a water sample here? Or
we're now on our third water sample, right?
We've already taken 3.
So it would be the 4th one.
It's up to you.
We've got, 2 hours left of our dive? Yeah, it's always a difficult call. We never know if we're going to come across an area with even more fish. I'd say we wait just a little bit more and see.
Okay.
Oh, look at that. You can see really how small they are.
Yeah. So does anyone know what kind of fish these might be?
So those lasers are just 10 centimeters apart. So most of these fish are going to be just like 4 centimeters long.
There's a few that are bigger, but not many. This is a really amazing shot of these fish. I would love to know what they are.
Thank you. Thank you, pilots. That's great.
That was amazing. And really, again, the camera that's on the Deep Discover ROV is really amazing. It can zoom it 18 times. And so you can see these details on these tiny, tiny creatures. And it's incredibly helpful for IDing. It's incredibly helpful to understand what the structure morphology of the biota is. It's also very helpful for the same things for the rocks when we get to zoom in and see what is in that biogenic sediment, what's on that rock. It's so helpful. Oh, and so now we're zooming in on the rock.
We have a little squat lobster walking sideways, and a couple of sponges there in the center, encrusting ones. One is actually a very pretty blue color, but these are really, really difficult or nearly impossible to identify just from images.
Right, and the tiny crinoid, the holoplus at the top.
Yeah, look at that. That's a small one, completely closed.
Also, look at the rock. If I squint my eyes enough, I might be able to tell you that I think that there are some horizontal striations that are coming down.
We can actually see that.
Yeah, so I see that. Do you see that?
Yeah, I see that as well.
And that may be some layering that, was when it was originally laid down, because we know that this is not in place. This is not where it originally was. So the fact that we have these dipping lineaments may have helped to discuss maybe where it came from. Very cool, though.
Yeah. And some more of those hydrocorals, really pretty. They are possibly in the genus Crypthelia. And that's, again, a genus that we have found on the Mid-Atlantic region on the Azores Plateau, even though these could be different species. So it's good to see some of these commonalities between such distant areas.
Right. You said Crypthelia?
Yeah.
Doesn't crypt mean like hidden or unknown?
Yeah, that's what it means.
So why is that called Crypthelia then?
I'm not sure. I don't know if it's because of the polyps that are sort of hiding inside those sort of capsules that they have towards one of the sides of the colony. So maybe that's the reason. But I would have to look it up.
I was just wondering.
So quite often, but you're right. I mean, quite often the names of species or genera are attributed on the basis of some morphological characteristic that they have.
Mm-hmm. We are so lucky to have seen this today. It's just so cool.
Yeah, it's really interesting.
And so what we've been seeing is that the outcrops are still few and far between, and they are probably out of place. They probably have, tumbled down from somewhere else, but they provide such important places for the biota to thrive. I do like this one a lot. If we were allowed to sample, I'd ask to take this home. What was that pink fish that was in the bottom corner?
It was very fast. Not sure that was a queen snapper again. If it was, it had slightly different coloration. It was not just that sort of red.
Mm-hmm.
Wow. Yeah, and here you see again, that's what you were talking about, these sort of layers.
And in sedimentary rock, which is what we're looking at, generally layers are laid down mostly horizontally. So we can pretty confidently say that this rock is not in place because this isn't how it would have formed when we look at it. And it is just completely crusted over with stuff.
Yes, we are. I think we can move.
All right, we'll say farewell to this beautiful outcrop. And then we will move up a little bit.
See what else we find. Yeah, we are now at 398 meters depth.
Yep. And so we're still climbing up this slope on this northern part of the Lang Bank. So we're actually facing south the whole time, right? Because we're on the northern slope, going south, moving up. And it's really been just a privilege to get to look at all these really exciting things on the seafloor. It's been an amazing echinoderm dive thus far, which none of us expected. But one of the things we are trying to look for is the fisheries and what fish we might be able to see.
And see if we find some evidence of areas or habitat that are particularly suitable for some fish, be them juveniles or adults, to aggregate. So do we find any evidence of spawning areas or nursery areas for some of these fish species?
What's that?
Oh, what is that? Oh, it's again some sort of root that we've seen earlier, or skeleton of possibly of a coral.
Don't know.
Yeah.
Is there a coral wrapped around this? In the bottom center?
There's
see the light colored?
There is. Oh, yes, there is. Oh, these look actually like Corymorphus.
What is Corymorphus?
So this is a really interesting group. We've seen them earlier, but not in these densities. Let me just have a look here at my notes, actually, because I think we've seen these earlier.
Are those the solitary hydroids?
So Corymorpha.
Yeah, it really looks like that.
What kind of coral is that?
But we've been mostly seeing those in the solitary, so not like this, aggregated like this. So that is a genus within the hydrozoan family, Corymorphida.
I don't remember seeing something like this before.
We saw a couple of them isolated, and they were pinkish in a previous dive. I don't think we've seen them on this dive.
Fascinating. So everybody in the chat room agrees.
Yeah, and here I think we have possibly some zoanthids, some very small zoanthids. Ooh. And again, here some brittle stars wrapped around whatever this structure is. We've really seen many, many different types of echinoderms throughout this dive.
We really have.
It's amazing.
And they're just so cool. So, I mean, it's pretty awesome that we've gotten to see however many of these that we've seen. I think the snake star might still be my favorite, though.
I think so, too. I agree.
But the Conolampus has such an interesting story behind it.
Yeah.
The sort of the traces it makes on the seafloor.
There's again here some more of these dark red urchins that we've seen earlier. Or maybe these are yet different.
These might be the ones that have the puffy, inflated...
Oh, no, these are not urchins. These are anemones.
Yeah, those are anemones. They're too wavy.
Yeah. From a distance, they look like the spines.
We talk about echinoderms. We think they are echinoderms. And so if you've just joined us this afternoon, we're here at Lang Bank. This is dive 7 of EX2206, the voyage to the ridge. We unfortunately had been not weathered out in the last 2 days, but sargassomed out.
Yeah.
Too much sargassum in the water for the ship to operate safely. But today we've moved to this location northeast of St. Croix on Lang Bank, and it's much nicer. There's little sargassum in the water. And it has allowed us to have what is already a fantastic dive with several hours left.
And we've been mostly doing a transect on the northern wall of the Lang Bank. We started roughly at 700 meters depth. And so we are now at roughly just 388 meters. Expect to be making still some way up to the 300 meters roughly. Yeah, and we see here actually another one of these glass sponges that we saw earlier, possibly in the family Rosalidae.
And look at this block. You can tell it's completely out of place. It's just a chunk of rock that's come tumbling down and is resting on another rock, but perhaps right underneath that block might be a fish.
Yeah, let's have a look at what's underneath it.
Pilots, could we zoom in on that?
I think we see a caudal fin there. Yeah, yes we do. And it's hiding.
I was going to say, I hope you can identify it from that. I can't.
Would be great if we would turn around. And to the right of it, actually, we see a tetractinellid sponge, a very spiky sponge there. Trying to see, maybe we can see it there from different angle.
That fish has got the right idea.
Ooh,
there's
actually
2 fish.
There's more.
Yeah. Good eye, pilot.
Yeah, we do see the eye there. Without seeing the completely, the head is difficult to identify. I don't know in terms of coloration. It could be one of those Darwin's slime heads, but
They seem a little bit skinnier than a roughy.
No, actually, we're getting an identification here from the folks in the chat room as cardinal snapper or muniama.
Okay, that makes sense.
Oh, so that's one that so maybe this is one of those that passed by very quickly once that had a mixed color.
Cardinal snapper. That's great to know.
Cardinal snapper.
And there were 2 of them.
And I would imagine this is one of those commercial species that are of economic relevance here in the area, which, as far as I understand, is very much based, on a small scale fisheries.
Mm-hmm. That's another one of those echinoderms.
Yes, it is. Look
Such
at that.
Interesting body plan.
Yeah.
Don't you just want to take one of those home?
I don't think you would be happy.
Probably not. Plus, we can't sample today. So, probably a good choice to leave them on the seafloor all the way around.
Is that again one of those snake stars, brittle stars?
It looks like it, but it's a different color.
It's a different color.
Yeah, the ones that we saw were much more like maroon and white,
yeah.
And this one is more like orange and yellow. So I wonder if it's the same or if it's something different.
Something different, perhaps. Again, we see here again one of these glass sponges with lots of spicules protruding out from the surface of the sponge and collecting all of that sediment.
Yeah. So Michelle Scherer, who's the one who identified the cardinal snapper in the chat room, she said that both the queen snapper and the cardinal snapper are part of snapper unit 2, but I don't know what snapper unit 2 means. So Michelle, maybe if you could put into the chat what that means and why it's important, that would be great to know.
Oh, here we have a better shot also of this
so that's
brittle star.
Yeah, so that's a management unit. The snapper unit 2 is a way to manage the fisheries. Thanks, Michelle. This does not look like, it's stripy like the one we saw before.
It looks different color.
But yeah.
Yeah, and Michelle here is adding here in the chat room that these are 2 of the most valuable fin fishes in the U.S. Caribbean.
The cardinal snapper and the queen snapper.
The queen snapper.
Mm-hmm. And so we're talking about the fish, but we're looking at this beautiful ophiuroid brittle star.
So it seems that Chris is also very excited about these ophiuroids.
I would love to know what he says it is. Yeah, it's quite beautiful. And just a reminder to all of people who are watching today, because we were within 12 nautical miles, we can't collect today. So instead what we're doing is our best to do some fantastic imagery that could be helpful. Oh, we managed to stump Chris Ma, our echinoderm expert. He is not entirely sure what the stripy ophiuroid is.
So it's good that we managed to get a good image of it.
Even our pilots think that if Chris doesn't know what it is,
it's special.
Yeah, what are we going to do?
Oh. And here we have one of these very long-stalked crinoids. Yeah, let's have a... Ooh. And here we're watching now actually 2 different sponges and in 2 different classes. So this one here is clearly a demo sponge, and it seems to have this crown of spicules that are surrounding the oscule, which is very unusual. This is something we mostly see in calcareous sponges. So we're trying to make here a little bit of way up slope now. And we are 377 meters depth. Another one of those glass sponges. Oh, in a coral here to the right. Another one of those black corals, possibly a Stichopathus. So we've seen a number of these during this dive. And we can see actually here now, the slope of the area that we're surveying. We can see here also to the left there is a very small white coral. Often it's very difficult actually to see them against this seafloor, this carbonate seafloor. Yeah. Yeah, and again it has a brittle star also associated with it. So this is a fan coral that we have seen earlier on and has that the snake brittle star. So this seems to be a pretty tight association here. So we've been finding these two together. So that's Asteroschema, the brittle star. Oh, is that a stalked crane? No, that's a very long stalk. It is. We're seeing a few more of these now on this part of the dive. Very long stalk. And so for those that may have joined us just now, we are here in the Lang Bank. We've started our dive quite a number of hours ago, roughly at 700 meters depth, and we've been going upslope on the north wall or north slope of the Lang Bank. And our goal is to get to approximately 300 meters depth. So we are just 9 nautical miles northeast St. Croix. And this bank is a very important area where a number of commercial fish species aggregate and spawn, namely the queen snapper and cardinal snapper, of which we've already seen some individuals. Oh, what is this fish doing? Oh, that's interesting. That's a strange behavior. Not sure if he's feeling well. How strange. No. Oh, strange. The color is fantastic, but yeah, it seems to be behaving awkward. Although what we know about fish behavior, maybe this is perfectly normal for him. But it is beautiful. It's very interesting that the top, it's very interesting because the top is very flat. So that's a bit unusual. Checking here if we get an ID from our experts here in the chat room as to which species this might be. Oh, it's spinning around. Okay, I think that's okay. Thank you, pilots. And so we've already come across a number of rocky outcrops or large carbonate boulders that have possibly come from further up in the Lang Bank. And these were typically more colonized by suspension feeders, filter feeders, such as corals and sponges. And they also seem to serve also as an area for where we've seen some fish species hiding underneath those rocks. Actually, just here in view, these are some rock sponges as well.
I was wondering about that.
Yeah.
They did look like rock sponges.
So before I do step out for a minute, I will just pass it on to Deb.
Who can't say anything about the rock sponges other than they are rock sponges. But we're at a depth of 354 meters and still just moving upslope. And we're just moving over top of this sponge. So I think this one's still living. It's covered in tissue. And that central area is where the water gets expelled. It comes in from all of the sides and it's expelled through the center. It does look like it has a similar morphology to some of the ones that we've seen earlier in the dive that were dead, but I'm not entirely sure it's exactly the same because this one is a rock sponge and I think the ones we had seen earlier were not. If only our sponge expert was still in the room, but... And then there's this small whip coral that we're passing by in the center left. It's white, it's pretty, just blends right into the seafloor. And what we found on this dive is that whenever we found some outcrops, not just hard bottom, but definitely outcrops, is when we've seen some of these very important commercially interesting fisheries species. Oh, what's that? It's moving. It's a crab with a really giant sponge on it. It's a brittle star? Oh, it's a brittle star. It looks like it's under a sponge. That's interesting. Looked like it was kind of flailing around down there, but it's probably quite happy. And so the seafloor still remains quite sedimented. And there's another brittle star. And this one's taking advantage of a little crevice, a little hard part of the seafloor. And look at this, how beautiful is that? So we just saw a brittle star a few minutes ago that had the same coloration, but it was banded. This one's quite beautiful. So it's another unknown in terms of the brittle stars. It's a beautiful coloration. I think we've given our Echinoderm expert, Chris Ma, a lot of work to do tonight. I think he's going to be up late trying to look up some of these species that we've been seeing. That one was quite amazing looking. We're just, again, moving up and seeing what there is to find. And this looks like a crinoid and a rock sponge behind it. And what's interesting to me is how these are managing to root in to the sedimented seafloor. I think there must be some hard bottom pieces that are underneath the sediment, but it's just surprising that they're managing to find such a successful place to live. Can we do a snap zoom on that? That must be a crinoid. Never mind. We've seen that before. There's a little sponge, I think a glass sponge in front of us. And so it's interesting because it's not like there's a huge amount of diversity, but we are seeing a number of creatures, and we're seeing a surprising diversity of those creatures. So it's not like we're necessarily seeing tons of things on the seafloor, but everywhere we look, we find more. And this is one of the things I really like most about working with the Okeanos Explorer is being able to explore the seafloor, being able to go to places that we've never been before and learn something new every time we get in the water. Yes, please. So there's another one of these. Maybe it's a pom-pom anemone? Maybe not. I don't know. Possibly someone will tell me. I feel fairly confident that it's an anemone with a little shrimp swimming by. Thank you. So one of the things we've seen as we've been driving up this slope today is that whenever we find a hard return in our sonar, which is when something is more reflective than the soft sediment around it, tends to be a rock outcrop, and we've tended to have a lot of good luck looking at the biodiversity there, looking at the fish species, looking at everything that's living on the rock. That is impressive. And here's another little outcrop. So we immediately see another crinoid with a brittle star and what looks like perhaps a sponge. This looks like a little ridge to some extent. So we're zooming in on this coral with a brittle star on it. Actually, with 2 brittle stars on it. Or more. No, many more. Lots of brittle stars on it. Wow, that's a lot of associates of the brittle stars on top of that coral. So we're continuing to move up and just see what we can see. There's a whip coral in front of us. This doesn't look like the most hospitable place for animals to live. It does look like there's a lot of sediment that's, although it is covering what is probably a little bit of a hard bottom environment. Oh, so that one that we were looking at that had all the anemones on it, the brittle stars on it, was an octocoral. We're seeing the isolated instances of sponges, many hanging out with brittle stars, a few pieces that are either extinct coral or old branches, we're not sure which. Is that another rock sponge? So, John, we had a question from the chat room scientists whether these large, roundish sponges are the same as the ones we had seen further in the dive that were dead.
No, I don't think so. They look different because I think we've seen some that we saw earlier in the dive. Some of the dead ones seem to be on that group that has a spongine fiber skeleton,
Yep.
so definitely not a rock sponge.
That is what I said.
Yeah.
That's what I thought.
See?
I know, I learned something today.
Yes.
That I thought that you had said these were rock sponges and those had the
Yeah.
spongin fibers instead and were not.
So I'm trying to figure out for these species. So this is a rock sponge, and I think it would be in the family Theonellidae. But I will have to I'm gonna look this up because if it is, it's in it's a family that we don't find so often in the Northeast Atlantic.
Very cool.
So it's a completely different group, and we know that the fauna that we find on the tropical Western Atlantic and on the Northeast Atlantic is very distinct, with only maybe a couple of species that are shared between these two subbasins. Oh, but this is really beautiful detail.
Awesome.
That we can.
And then the brittle star that we're seeing below, we've now seen this a couple of different times, this orange and yellow banded brittle star.
Yeah.
But when you were out, we actually saw another orange and yellow brittle star that, instead of banding, had like long stripes along its body.
Along the body.
And yeah, it was another one. We've managed to stump Chris Ma, our echinoderm expert, a couple of times now. So
and that's quite
both of you have homework.
and that's quite impressive. Yeah, both of us have homework to do now. And we know for a fact that this tropical Western Atlantic is a hotspot for these lithistid sponges. So we find way more of these species here in the tropical Western Atlantic than we do on the Northeast Atlantic, and then even less so in the Mediterranean. So this must have to do, with their evolutionary history and the biogeographic patterns of those the group as a whole.
And that is one of the things that we're trying to study here on the Voyage to the Ridge expedition and the larger Aspire campaign under which this falls, is trying to understand things like connectivity, trying to understand why certain species would be in certain places and why they wouldn't, and where the range extensions might be. So each piece of information that we collect, even through imagery, will help us to understand that a little bit better. So one thing we had really hoped to see on this dive was fish or juvenile fish, because Lang Bank is known to be an area where spawning occurs, and we wanted to see if we might find some locations where these commercially important species like queen snapper are spawning or even living. Oh, that brittle star is trying to get away. Run, brittle star, run!
Go.
It's like it's running in slow motion. Oh, and now it stopped. And so here we are on dive 7 of EX2206, which is one of the series of expeditions for Voyage to the Ridge. We've had 2 days where the sargassum was so bad that we weren't able to dive, but we found a nice location today right off of St. Croix at Lang Bank. And we're looking at another one of these crinoids. We've seen these a couple of times already on this dive alone, and they're very interesting because it's a stalked crinoid, but it can kind of walk away if it wishes to, is my understanding.
Yeah, that's correct. And we've seen also that, or we heard from Chrisma earlier on, that sometimes some of those Citroid Urchins actually feed prey on these, especially at the base. Oh, look at that.
Wow.
So it has this sort of it looks a little bit like a root system, but so apparently it can just release that and just...
It can go walk about?
Yeah, fly away. Or swim away.
And we're still seeing that the sediment here, has a lot of biogenic material in it. There's a lot of coral rubble. There's a lot of... where's a fish? Oh, there it is. Is it a fish? Love the fact that you say the word fish and we all stop talking and look for fish. Could we try to zoom in on it? Oh, yeah, look at that.
Almost looks like a rock
What
fish.
A fantastic eye, pilot.
Yeah, looks like one of these rock fishes, doesn't it?
When we're done zooming this fish, could we look just directly below it? There are some pink things in the rubble, and I would love to know what they are. Oh, that fish is awesome looking.
Yeah, look at that.
I love those eyes.
Yeah. We've been seeing quite a number of grumpy fish around.
I think I often look like the grumpy fish. No, it's a beautiful fish.
It is. Look at the beautiful iridescent color of the eye.
Do you know what kind of fish this might be, Joanna?
I think this is a what I call a rockfish, so like these scorpionid fishes. But let's see if someone here in the chat room can give us a more detailed ID.
So I'm totally curious to know what these pink things are in the sediment.
Let's have a look. I wonder if these are coralline algae. I think they are, probably. These rhodoliths of coralline algae. Yeah.
That's a great view.
Yeah, I think that's what these are.
Okay.
So these are calcareous algae. They have typically this coloration. And they build over the course also of many, many years. And they don't look like algae at all because they are, this sort of calcareous wall.
But that also gave us a great view, not just of the coral and algae, which thank you for doing that. That was great. You could also see the broken pieces of coral. You could see broken pieces of shell.
So very biogenic sediment here.
Do we want to zoom in on whatever is on the left that's yellow?
Yellow, yeah. Could have a quick look at that.
And I'll just note that we still do have 2 of our water samples available, and we have a little bit over an hour left of our dive, I believe.
So we should get those filled relatively soon.
Are those encrusting sponges?
Yeah, those yellow dots are some encrusting sponges. And here to the top right, we have a number of these serpulid worms that build these calcareous tubes, and then the worm, or bristle worms, and then we see the worm, sticking out from the tip of those tubes. So these are cebulids.
And so we're at 325 meters already. We started at around 700. So we've been moving consistently up this slope.
Yeah. So we've made quite some progress.
Yeah, into what I would consider shallow water.
Yeah. And we see here again one of these large rock sponges. Yeah, it looks like a fishing line there in the... Yeah.
On the top left-hand corner?
Yeah.
Ooh.
It's a long one.
It is.
And of course we have to be very careful when we encounter something like this.
That's a lot of line.
Yeah. Yeah, these long-stalked crinoids seem to be rather common in the area.
So we're just going to hop over this fishing line and get out of its way.
Oh, there! I think there's a fish there
Yep,
coming into
right in
view
the
now.
center.
Yeah, right in the center. Let's have a look at that. See if we can.
Let's take a look at the fish first, if we could.
Yeah. Oh, it looks. It went into its burrow. Look at that.
Oh.
Yeah. There's a couple smaller ones here to the left now. So actually we are starting to see a little bit more fish. Small ones, possibly juveniles.
But yeah, they're all darting around.
But yeah, but the larger one really got underneath that carbonate crust there.
Oh, that's the reason they made it to be that old. They know to hide.
Yeah. So it's interesting to see that perhaps these carbonate crusts that have this, this hollow interior, may be serving as an habitat for this juvenile fish here.
It makes a lot of sense.
Yeah.
What is that in the middle, right near that fish? Is that a sea cucumber?
No, that's actually an
is it a worm?
A worm.
How cool!
It looks like these fireworms.
Oh,
Hermodisa. And we call them fireworm because they will sting you, and it will be painful. It will feel like you've burnt yourself.
Delightful. Yep. So that's a fireworm.
It really looks like it. Yeah.
Yep. You got a confirmation in the chat room.
Yeah.
And it was bigger than the fish that swam by it, or just about the same size. Those fish are not big.
And to be honest, I'm not sure how common these are in deep sea. Remember seeing a lot of them in shallower waters in the Azores archipelago while scuba diving, and we learned early on that we should not touch these.
I believe one of my friends did her thesis on fireworms.
Really? So the species we have there is Hermodisa carunculata. I'm not sure if this is the same species, and they can grow actually relatively large. Yeah, and Scott here is making a very valid comment, saying that they will sting you is actually a false impression. Indeed, it's just that those bristles that extend from this fireworm have a toxin, and so when we try to grab them, of course, we will sting ourselves on it.
But they're not actively stinging you.
So it's not exactly. They won't actively sting you. You should just stay away.
So what kind of whip coral is that one?
We couldn't see. It was relatively fast, but most likely a Stichopathus black coral. Oh, not a black coral. Okay. So maybe that was a different one.
Elicellid whip?
So apparently it was not a black coral, that one.
That's interesting.
I didn't look at it in detail. So that, okay, so there's another one here actually coming into view now.
Those are very hard to see against the white, the light brown sediment.
Yeah, it's true. So yeah, so this is actually different. It's a different. It also has like sort of this whip shape. But so apparently this is an Elicellide in the Elicellide family. So it's an octocoral, but not black coral as we've seen earlier. But it does have another bristle star on it. It seems to be quite long as well, this one. And so we're now here at 313 meters depth. So getting very close to our target depth of 300 meters.
Can we go further up?
Better start dialing.
Oh, and we
Chris.
have a different sea star props here, right center. Or maybe it's, I think we may have seen actually this sea star before, one of these goniasterid sea stars earlier in this dive.
Sorry, can we get a snap zoom on that sea star? There was particular interest from the science community in the colors. Thanks, pilot. Yeah, we wanted a snap zoom on this.
What is it sitting on top of?
It's difficult to say if it's right on top of the carbonate crust or if there's something
I think there's
in
something
between
under,
and
yeah, I think there's
underneath
something
it.
underneath it.
I see there to the lower left a little bit of a yellow encrusting sponge,
That's
but
what I
I'm
was
not.
looking at.
But I'm not sure if there is that is the same that is sitting right underneath it. So it could potentially be feeding, but it's it's really difficult to say.
That's what Chris Moss says it is. It's feeding.
Yeah.
It's looking for dinner.
So it's just going around and see what it can find. Maybe that small encrusting sponge was not didn't look appetizing enough.
You don't know. Maybe it was once a large encrusting
Or
sponge
maybe, yeah.
and is now a small encrusting sponge.
That's also true.
You don't know. I never, up until this this expedition, I didn't know that echinoderms, and especially sea stars, ate sponges. This is a really exciting development for me.
Oh, and maybe we could have also a snap zoom here on the coral. I wonder why part of it seems to have the skeleton far more exposed than other parts here.
So this is very interesting. One of the scientists in the chat room, Steve Oskovitz, says that the deepest depth that they recorded the photosynthetic organisms of the coral and algae was 256 meters in the Anegada Passage.
And we are in...
And we were deeper than that when we were looking at the coral and algae, or what we thought was coral and algae.
We're considerably deeper, yes. We're probably at 320 meters depth or so. We didn't see much of it. We only saw just a few fragments.
But we have been seeing them. I saw them before that, too. I just didn't have a chance to zoom in. Yeah, we were at 326 meters when we saw that coral and algae.
Yeah.
That's very interesting.
And perhaps we could also, at some point, then zoom in on those red spots we saw there, but we can come back to that when we see more of it. Yeah, but we passed it, so that's okay. I'm sure we're going to come across more of it. Thank you.
So what is this?
I'll have a look at that.
What is that?
I'm trying to figure it out.
It looked like a sea star that dissolved.
Yeah, it does not look like a sponge to me, but let's maybe if we can have a closer zoom into it. I'm not seeing any.
Does it have a bacterial mat growing on it? Is it possibly
like
something recently dead? No?
Not sure, to be honest. No, it doesn't. It doesn't look like it's dead.
Okay.
But it doesn't look very spongy to me. But that's actually a very good question. I do not know if this is a sponge. It does not look like a sponge to me. I don't see any clear openings or, water canals going through it.
I'm going to go with not a rock. Not a rock. Hey, but is that the bulbous sponge to your left?
Yeah, that one is a sponge. It could be in one of those groups like Oceanapia that have these long fistules. But there are a number of other genera, actually, where we can find this.
And Pilot, there's a request that if we can see the little pink fishes that went by again, if you could try to zoom on them, that would be great.
And we have here a large anemone here at the bottom.
Me neither. Okay, so what is that at the bottom?
Yeah, that's an anemone.
That's very colorful.
Yeah. And I think here we're really starting to see, a bit of a shift in the species composition. Some of the things we're looking at now look no more typical of shallower or relatively shallower water. So I think we will be soon entering this mesophotic area.
I mean, it makes sense. We're at 310 meters. We are substantially
shallower than we were at the beginning. Yeah.
So if we can see the little pink fishes, we're going to try to zoom in on them.
Okay.
But I haven't yet seen them personally. No. So that's an anemone,
beautiful.
Huh? With all of its
beautiful
tentacles retract, right? That is really cool.
We can see here they have these more robust tentacles. It's very interesting, and they have also these stripes along the tentacles. And there in the center, it's its mouth.
So we've got 45 minutes left on Dive 7 to Lang Bank. We've been having a wonderful time today. We've really managed to see a lot of very interesting species. I think less fish than we had expected, perhaps, but so many more Echinoderms. So it's been really interesting, and this last 45 minutes is proving no different.
That's a beautiful shrimp there as well. So no more signs of those pink fish, I guess.
Okay. I hadn't actually seen them, but one of our shoreside scientists, Michelle, had pointed them out. Anyone want to take a last guess on what the squishy pink thing at the top is? Anyone? Anyone?
I'm starting to think whether it could actually be a coral. One of those most encrusting corals that are more typical in shallower waters, but
I don't know. It just looked like a pile of pink goo to me.
I'm not 100% sure. I have to say, it did not look like a sponge to me.
pink goo with spines and what look like white bacterial mat. That's about as good as I get for that one.
Yeah, and I was really hoping it would come across an area with more fish so that we could have a water sample collected.
Oh, we may still yet. Hope springs eternal. Is that another sponge in our upper right? Or is that something different?
Let's have a look.
Or is it an urchin? There's a small glass sponge over to the left, right? Rock sponge?
Let's have a look.
Let's have a look. We'll get a closer look.
Oh, it seems like something is there. That there's. It's again a brittle star underneath.
That is so weird.
It's not the first.
Because it looks like they look like they're trapped, but obviously they have chosen to be there for safety.
Yeah.
The arms of the brittle star flailing around the outer edge just makes me laugh.
It looks like it has just dropped on top of it.
Yes, exactly.
Let's have a look at this. Again, orange. We can have a quick look at this orange thing there in the center. And so now that to me looks more like a sponge. And different from what we saw earlier on. Indeed, yeah. So this one does look to me much more like a sponge, a demo sponge. Not quite certain of which group this would be. It's a heteroscleromorph. It reminds me somewhat of the genus Agellas. But so these are now really things that are much more typical of mesophotic areas, so not very, very deep sea. Oh, look at this urchin.
So that looks like, it looks like from the body plan, from the, I feel like we've seen this one before, but not on this dive. Maybe I'm wrong.
I think we've seen one of these.
Yeah. It's got those long, flexible spines.
Exactly. Earlier on. It's just incredible how many different urchin species we've just seen on this dive.
Aspito,
Aspito, Diatomated.
And yeah, so we did see one before that was more of a greenish, grayish color.
Yeah, and they're just to the back and to the right of it. You see what seems to be one of those coralline algae, a little bit of one of those rhodolites, but nearly white, which means that it's, pretty much dying or dead already.
But we have seen a number of it.
So it could be that some of these are actually coming
from upslope.
So they wouldn't be naturally living here because the amount, of light down here would be so reduced for them to photosynthesize that what's most likely is that they've been washed down from upslope.
That makes sense.
And then typically in shallower areas you will find, they will cover great extents of the seafloor, these rhodolite beds.
Well, then I'm glad I asked about the weird pink thing on the seafloor. And then you asked about the other weird pink thing on the seafloor, and we haven't figured out what that one is yet.
Oh.
So there's a stalk in the top right corner.
Yeah.
So we're just coming up on the 300-meter contour isobath. It's very shallow here.
Oh, so we just made it to the 300 meters.
Yep.
Oh, there's one of those stalked crinoids seem to be, just bent down. I wonder what it's doing.
Probably wondering what we're doing. Go ahead, Nav. We will let you know. We will talk about it right now.
I think we will have to take one of the Niskin bottles soon, since we're getting, even though we haven't come across more fish aggregations here.
It's up to you. I mean, we don't have to take all 5 of them if we have that option of not.
Yeah, but we're just
What is this crinoid doing?
Yeah, it almost looks like it's getting rid of the brittle star. Look at that.
Yeah.
Look.
Chasing the star.
It is.
Well, that's interesting behavior.
Yeah. It's fighting back.
Oh my god.
Oh my gosh, that's crazy.
That's so cool. He is very unhappy with this brittle star.
Yeah, it really looks like it's sort of fighting it off, like, get off my my stalk.
Get off my stalk.
Wow, very interesting. Look at that.
We could keep watching this. This is great.
Yeah.
I wonder if this is a behavior that's been seen before.
Oh, and
wow, it's maybe successful. Yeah.
Let's see if the brittle star is really going to go away now. Oh, and it will.
I'm going to go find something easier to eat.
Wow.
Look at that. This is really spectacular.
So you get to watch something very enjoyable in terms of behavior with this crinoid and this Ophioid. And that might be a novel behavioral thing that we've seen.
Get off my lawn.
Yeah, exactly. Hey, kid, get off my lawn. But it certainly has drawn some interest from the folks in the chat room as well.
Yeah.
But I think the brittle star didn't quit yet. She's still just hanging there.
Coming back for round 2.
Coming exactly.
Or 4 or 12. We don't know.
This is really interesting.
What's that shell in the top left-hand corner? Okay. So I think we heard that there's another area of somewhat high reflectance just a little bit ahead of us.
Oh,
and again, that harder reflectance might be an outcrop.
One of those carbonate outcrops, which have been very interesting. And here we have another sponge in the class Demospongia. This reminds me a lot of a Geodia sponge, actually, that we also have in the Northeast Atlantic.
We collected one on one of our dives in the Azores, right?
Yeah.
We've been very lucky to see such a great variety of environments on this expedition.
Yeah, that's true. All the way from
mid-ocean ridge,
mid-ocean ridge, fracture zones, sea mounts, insular shelves, and slopes.
The Azores Plateau.
Yeah.
So much to look at there.
I guess that will be Chris.
I hope so.
I hate to disappoint you all, but
it's Scott.
That's
hey.
Okay.
That's quite all right.
I'm sorry you already gave it away. Calling in for the end of the dive.
Oh, Scott, what were you going to tell us about, though?
I think Scott was preemptively calling in for the dive planning call.
Oh.
Yeah, I'm just trying to, get conditions so that we come across a bamboo coral. I'm being really hopeful, but then, just ready for the dive planning call.
Okay.
Yeah. Thank you, Scott. Unfortunately, I don't think we came across any bamboo coral on this dive. We did see. We did see a couple on our previous dive, though.
Mm-hmm. But we can keep hoping.
Yep, I'm going to keep hoping. This is the first time I've been able to call in all day, so sorry I couldn't be in there.
We are glad to have you on the line. I just love how these rock sponges, these demo sponges, are sort of thrown everywhere. They're sort of here and there, scattered upon the seafloor. So it looks like a small rock outcrop ahead of us.
Ahead of us there.
Yeah. And that was probably what they were seeing on the sonar, which helps to look at soft versus hard reflections. So often it can show a rock outcrop in the midst of a soft sediment. And whatever this was, it's pretty sedimented now.
Let's see if we see some fish hanging out underneath that fragment there.
Mm-hmm.
Oh, and I think that's again one of those very large anemones there. Oh,
what is
and
that yellow thing?
That's another sponge, and that must have certainly stumbled down from
earlier. Let's try to look at the fish.
Yeah.
Yes.
Oh, look,
little pink fish.
Those are the pink fish, possibly. Okay. Oh, there it is. Yeah,
I'm not sure,
probably.
but it is a pink fish.
Yeah.
What other exciting things might we find under cover of a rocky outcrop?
Yeah, let's have a look at it. Oh yeah, this would most certainly not, and you can see actually part of it actually it still seems to be living, but most of it you can see already the skeleton, the fibrous skeleton exposed.
So the right side and the bottom side are still alive,
yeah,
right?
the right side still looks like it's still living, whereas this part that we're zooming in now, you can see these spongine fibers really exposed, meaning that all of the organic part of the sponge has now been largely dissolved.
It looks a lot like my kitchen sponge.
Yeah. So this would have most certainly come from an higher area here. So this is a dendroceratid sponge. So it's a group that has these spongin fibers. And I'm just looking here quickly for name for this species.
And the fish that we were looking at, the little pink fish. Oh. Go ahead, Scott.
Sorry, I was saying I'm not sure if the Serianthid anemone at the base.
I think you mentioned something about the anemone. You're coming in a little bit faint. But
I said I think that might be a Serianthid anemone, a tube anemone.
Thank you. That was much better. So yeah, Serianthid anemone that we were looking at when we were looking at the sponge that had come from a higher depth, lower depth, shallower depth. I'll go with shallower depth. High and low don't work very well there. And Joanna, what are we looking at right here?
Just a moment here, because I was. So was this one of those? Yeah, was this one of those weep corals that we've seen earlier on?
Scott just said it was an octocoral.
Right. Yeah. Yeah. So it may be possibly we had suggested, or I had suggested earlier, in the family Eliselidae, just because that's a pretty common family in the kind of this twilight zone. So from about maybe 80 meters down to about 300 meters, and they're definitely stocked ones. I'm not very familiar with them in situ. But that's definitely a good bet.
Yeah, and so I think actually that yellow sponge was possibly in the genus Aplycina. It's a genus that is way more diverse here in the tropical Western Atlantic than it is on the Northeast Atlantic, where we only have a couple of species. One of them is Aplycina aerophoba, which is also yellow like this one, but it's much smaller. It also has this tubular shape, but is typically much smaller than the ones that we find in the tropical and Western Atlantic.
Yeah, a short snap on it would probably be helpful for identification now that we've seen it 3 times.
Oh, actually, this looks different from what we've seen earlier. This sea star. If I'd have to guess, I would say again a Goniastrid sea star, but I could be wrong. This has a much darker coloration of these spots throughout the body.
Textures are just remarkable.
Yeah.
It's the extreme close-up of the sea star.
Yeah. Oh, and so this seems to be another one of those Anthanoides perisae. And I hope Chris is referring to the sea star, because we've seen now so many different species. So many different names. Yeah, and Chris is saying here that this is just a larger
Did you have
one,
lasers
and then
on
maybe
it?
there's color variation. No, actually, pilots, can we turn the lasers on just for scale? Oh, yeah.
Oh, look at that.
Look.
Its feet are perfectly 10 inches, 10 centimeters apart.
Yeah.
Perfect. Oh, it's sliding.
Oh, it is. Oh, I wonder if there's something living there underneath.
It looks like it.
Can we have a zoom in on that opening?
Oh, it looks like an eyeball.
Yeah, it looks like one of those anemones.
Yeah, it does look like an anemone.
Yeah, I was hoping there was some fish hiding there.
Does anyone else see a face here, or is it just me?
I see a face.
Okay, good.
Two eyes.
Get a little worried. Two eyes,
two eyes, a nose.
Yeah.
Wearing some sort of hat.
And there's something in each of the eyes. The anemone is on the right side, but what's on the left?
I think it's also another one of those large anemones.
If you look back,
and there's one of those pink fish.
If you look back 4 minutes into the chat room, you will see that I said deep sea nostrils.
Deep sea nostrils. That's a good one, Scott.
Oh yeah, it's another one of the two. It looks the same.
Yeah.
I try not to anthropomorphize the seafloor too much, but this did look like a face.
It did look like. Oh, look, a little fish there.
Oh my God!
No.
Oh my God!
Oh
no!
Poor fish. No way. We did not want to see this.
Oh no! Wow.
Oh no.
This is life in the ocean.
Boy, I can't wait to see the replay of that.
I think that anemone is now
happy,
and that fish probably not so much.
No.
Wow.
We were not expecting to see this.
Is that its tail still sticking out of the end?
Oh no, poor fish.
Wow. There's nothing else to say at this moment. All of us are just staring at the anemone.
We were not expecting this.
I'm not gonna lie. I can't wait to see the replay.
Well, the anemone needs to eat.
Yes, it does.
Wow.
I hope that wasn't a commercially important fish.
Oh my.
All right. Now I have questions about anemone digestion and what.
For how long will it be? Munching on that. I really don't know.
I think the moral of the story is to stay away from anemones.
Yeah. Gosh.
Well, I mean, we have to have a food chain, right?
Yeah, exactly. So that's part of the
sustaining all these organisms.
Yeah, part of the trophic food web.
To answer Thomas's question, yeah, exactly. So Thomas was, saying, how long the digestion. So I don't know, but it's interesting to hear you say munching, because there's no munching going on. There's basically a cavity that's kind of like a sac, and that fish will now sit in the sac. There are stinging cells inside that sac, so it could paralyze the fish. But otherwise, there's just a lot of enzymes that are going to be released into that sac, and so it's a relatively slow process because it doesn't have anything like teeth, and so it's just got to digest it now and there. So it could probably take a very long period of time. And the other interesting thing is it's probably not going to eat again until it digests it because it has to remove the undigested contents through that same opening. There's only one opening into the gut. So cool stuff.
So does it basically just dissolve the fish over time?
Yeah.
Yep.
Oh, wow.
Yep. And ultimately the final products of the digestion take place within the cells that are lining the cavity of that gastrovascular space. So it's got to be... Fish bits have to be dissolved enough that they can be taken up by individual cells.
Yeah.
Wow.
Yeah, so that anemone just got lucky today, and we'll have food for the possibly coming weeks.
Mm-hmm.
Oh, and this is again a fragment of a sponge that has probably come from further up. It doesn't seem to be really in place.
Mm-hmm.
And this is a demo sponge.
So what depth would those normally be at? We're at 278 meters right now.
I would say, of course, we don't know a lot about this mesophotic, the diversity in these mesophotic areas. or at least I don't know a lot of the mesophotic species here in the Caribbean Sea, but I would say that some of these species will most likely occur down to 70, 100 meters depth.
Okay.
And you could see that that one was not even attached to anything. It was.
Right.
It seemed to have just rolled down from upslope.
Yeah, the word you said, fragment, that really, that's what it seemed like.
Yeah. Oh, and we're getting an ID here for that small pink fish that could be in the genus Nymphaster, so slope fishes. That was really impressive.
I think we're all a bit shell-shocked back here.
We are.
I kind of hope we don't have fish for dinner now.
I think it really is the first time I see an anemone eating a fish like that.
It's just amazing how fast
it was
that
super
happened.
Fast, exactly.
What is that?
If I can offer just a little bit more about that anemone that we just saw feeding,
Mm-hmm.
then I'll let you talk about this red thing first if you want.
Well, I have nothing to say about the red thing,
I also,
so...
to be honest, I also don't know exactly what this is.
So, Scott, you can talk about whatever you'd like to.
Yeah.
Yeah, I'm studying this thing to see, is it like a single polyp? Is it a little...
No,
I
we
mean,
don't...
Joanna, obviously
no.
you don't think it's a sponge.
No, I don't think it is a sponge.
Oops.
So...
Yeah.
But it doesn't, we can't see, any skeleton on it either that would look like a solitary coral or something. So it's,
Exactly.
rather unusual.
I wonder, could it be a coralline algae? I don't know that they have a structure like that.
Yeah, I'm not 100% sure. Normally see the coralline algae more like this globular shape or plate, either encrusting something or this more globular rhodolite shape. So, yeah, I'm not sure.
It looks like a red version of the Guccisamia, that bright yellow coral that we see in the Mid-Atlantic
Yeah, but
ridge.
we don't see any of the tentacles, right? Yeah.
Correct. Yeah, they would have to be withdrawn. So, okay, so that gets back to the anemone. What I was going to say, we're seeing so many corals and anemones, and they all have their tentacles sitting out, and they're all basically just waiting for something to drift by and intercept those tentacles. And then when something does, it's that fast reaction like we just saw with that anemone, where it's, okay, finally, here's some food. I got to take advantage and close up. So essentially, we're all seeing, spring-loaded traps most of the time. And very rarely do we actually get to see them deployed.
That's very cool. It's a really good point, Scott.
Yeah.
What are all these things on the seafloor?
Let's have a look here. Actually, this again looks like that red thing we just looked at earlier on. It's really interesting because, if this is a sponge, it certainly is not anything like I've seen before, which would not be totally surprising because...
Just insert coral into that sentence, and I would say the same thing.
But it would not be surprising in the sense that, the tropical Western Atlantic is acknowledged as, biodiversity hotspot, and a lot of different species that occur here that also do not occur elsewhere. Or at least we don't expect to see a lot of these species.
sorry, a suggestion from Tina and Asako that it might be an octocoral.
Okay.
To check that out.
Is a cup coral a type of octocoral?
Been withdrawn.
Or is it different?
No, a cup coral is a stony coral. It's scleractinian.
Okay.
So that would be different.
Still catching up on my corals.
So am I, Deb.
Problem. I do have a blog on somewhere on the website that compares all the different kinds of corals, that general term coral, and breaks them up for you.
Oh, I'll have to look into that. It's always fun. I learn so much when I'm on these expeditions, whether on shore or at home. So you got a little fish next to a sponge?
Next to a sponge, yes. Yep. So that is definitely a sponge, and you can see some of those papillae.
Yeah, maybe zoom in on the fish first.
Very small, yeah. Very small ones. Oh, look at that fish. Beautiful.
Go ahead, Nev.
Mm-hmm.
That's a good question. Let me find out.
Yeah.
So
should we?
We could take one now.
Yeah,
I'd
say we take one last water sample here.
And then, do you want to take one in the deep scattering layer when we go up? Or we're already past it, actually, aren't we?
I think we've already passed it.
Yeah.
Actually, we took the first on the way down. So if we can take one water sample now, that would be great. And I think we'll close our water collections for the dive today. Actually, have you noticed this mark also on the seafloor that has been great? Thank you, pilots. So we've got now our fourth water sample for eDNA. Have you noticed there's like this deeper mark on the seafloor, and we've been going along this. I wonder what could have created that.
I mean, it might just be a little runnel where something slid down preferentially right there, because you are seeing sort of lightly striated seafloor, and it's probably just where things have rolled down the slope. And so we've got about 10 minutes left on the seafloor. And I want to thank everybody on the GFOE team, the ROV folks, and our video people who are fantastic. And, you know, I'll mention it again. We were talking about how great the engineers are, but we probably should have said everybody on the ship is really great. This ship does not work without all the people who are on it. You know, from the cooks and the stewards to the engineers to the deck folks who make sure that everything is running perfectly.
All the operations run smoothly. Yeah.
And, of course, to the NOAA Corps who are, you know, making sure the ship goes in the right places. So
with all the planning and mapping.
Yep. It's a really great group of people who are all committed to making ocean exploration happen.
Yeah, it really is. Yeah, we're seeing actually here a few more of those rock sponges, so what we call the lithistid sponges. There's actually a number of lithistid sponges here in the area that, although they have been observed and partly examined, they are still undescribed. So there is a lot of undescribed diversity for these lithistid sponges.
So as we're coming to the end of our dive track, we're getting a really nice view here of how steep the slope is. It's kind of just been curving upwards, steeper and steeper as we've gone along. But at the top, there's this flat, shallow area, about 50 meters deep, And that is a known spawning ground for a number of commercial fishes. And so part of the investigation we were trying here today was to see, do they come down the sides? Do they hang out on the in the deeper areas? And what kind of life and other biodiversity approaches those shallow banks that support those critical ecosystems?
But we are about 200 meters below that 50-meter area, so we're still, much deeper than those fish might be interested in. And
we may have just had here record depth for the queen snapper. We will have to confirm that with our shoreside scientists. But we had a record at 499 meters for the queen snapper.
So this was a really interesting dive.
It really was.
It's funny because I would say that in terms of abundance, it was still fairly sparse, but everything we saw was new and exciting.
Yeah, there was a lot of diversity.
And how fitting is it that we're going to end with an urchin?
Very fitting. This was definitely the echinoderm dive.
This was the echinoderm dive.
And it wouldn't surprise me that this was yet another species that we hadn't seen before in the dive. It looks like a small Citroid Urchin, but not sure if the same species that we've seen earlier on.
And its spines seem a little thinner than some of the others that we've seen.
Very yellow.
And so that's a... Oh, you were going to have to pronounce this. I can't even try.
Calocidaris micans.
Of course. I would have said that.
So we're getting here an ID from Chrisma.
And this is the kind of thing that likes to eat the stalked crinoids. Of course, now we know they fight back.
Yeah. So perhaps on our next dive we'll have a Citroid Urchin stalked crinoid match.
I mean, it really is amazing. It's when we're not just encountering the species and seeing them in their environment, but then seeing them do something behaviorally that hasn't been seen before is pretty cool.
It is.
Yep, and so that's about, 30 centimeters tip to tip in terms of its scales. Maybe it's actually 25.
Yeah, 25 or so.
Look at how well it blends into the seafloor, though. And so there's an identification of the little pink thumb-like thing. Bathyalcyon, which we saw about 10 minutes ago.
You mean the thing we weren't sure if it was a sponge or a coral?
We weren't sure whatever it was. It looked like a pink thumb.
Bathyalcyon. Okay, so we should look this up.
So again, just want to thank our pilots and everybody on the ship that supports ocean exploration. This was a fantastic dive. We really appreciate it. And also we thank the Sargassum for not coming by today.
Yeah, and all our shoreside scientists who really make these all of these dives just so much more interesting and exciting just by sharing all their knowledge on their groups of expertise.
Oh my gosh.
I was hoping that this could be the bamboo coral that Scott so much wanted to see, but I don't think it is.
That's very nice of you to at least have that, desire. So I appreciate that.
and now we're hearing that maybe it's not a Bathyalcyon after all.
Okay.
So
we'll
the
have
pink thumb is still unknown. Unknown.
Yeah, I think we're saying that's the best guess right now. It's
Okay.
possibly something related to that. Certainly, I don't know because I haven't seen them often enough in situ to know what they look like. But it's a reasonable guess because that's a large single polyp octocoral, and there are very, very few species known that have only a single polyp.
Okay, that's really interesting. Thank you, Scott. And one final look here also at one of these anemones. And we see again a few of these rhodolites.
Like the one we saw under the sponge before.
And we see actually also here some of these, some little bits of these rhodolite calcareous algae. There was another one here to the lower left. And there to the right, actually. And
that one to the left there, Joanna, looks a bit like what we couldn't identify before. And this time I'm seeing very clear circles that look like
but that one,
that
but I think that's actually on a different.
Perhaps you were correct that was some encrusting coral.
Yeah. Okay, and I think with this we're getting off bottom now.
Yep.
Once again, we'd like to thank everyone who has joined us from shore, both in the chat room and also just joining us through the live stream. And we will soon be having our dive planning call.
Yep,
thank you all.
Yep, we'll take 5 minutes and
sure.
Come back to you soon for the dive planning call. Thanks.